All right, looks like we are live on Standing for Truth. My name is Donny, and I am your host for this important video. I am here with my co-host as well, Matt Man, and we are going to be touching on topics. We're going to be dealing with topics related to human evolution, specifically the human-chimpanzee DNA similarity argument. We're also going to be getting into mitochondrial Eve, Y chromosome Noah, DNA function. We are going to address some criticisms that we've seen come in. And we are also going to be debunking a video. It's a short by an atheist YouTuber named Forrest Valkai. If I'm saying his last name right, I'm not sure. But he's got a video. It's a really weak video. It's soft. And so me and Matt are going to have some fun debunking that. It's a YouTube short titled Genetic Adam and Eve. And so this evolutionist who, yes, believes he's related to banana plants and pine trees, he puts forth about 30 seconds worth of pretty bad arguments, Matt. And, we could probably deal with that 30 seconds for about 30 hours. And so we're going to have some fun with that. Matt, what's going on, brother? Let's hand it to you first. This is, to the audience, this is live with Donny and Matt. So it's a little more free-flowing. It's a little more impromptu. We have discussed, obviously, beforehand what we want to get into, what we want to touch on. But in what specific order, we don't necessarily know. When we do these live shows, we did one a couple weeks ago where we responded to our favorite evolutionist, Gutsick Gibbon, on chromosome 2 fusion and nested hierarchies. And we engaged the audience. We touched on a number of topics. And so we're going to do the same here. It's going to be a little more laid back, but it will be about a couple hours. And we're going to touch on a number of things. And so, Matt, brother, what's going on? How you doing? Nice shirt. And also, what have you been working on lately?
Doing good, man. I've been working on a project. It's turning into a book now. It was an article size, and it's about radiometric dating. It's the one thing that young Earth creationists kind of either have one particular argument for, and there's just a handful. And I thought I would come at this from a different angle and give people an entirely new argument, and then we're going to be utilizing it for our side from now on. So I have a debate on it coming up, so you guys will get to see everything that I'm going to present in that debate, and then we'll test how strong my conclusions are. So that'll be really fun. I've been working a lot on that.
It's going to be some free peer review, Matt. We get to see you in the ring. We get to see you in action twice in June. And so you'll be debating Grayson from the Base Theory YouTube channel on the age of the Earth. And so you'll be testing your radiometric dating model and your various solutions to the heat problem. So that's going to be good. I'm looking forward to that. The audience should as well. Then you're also going to be debating Mark Reed at the end of June. So I'm keeping you busy, brother. That one will be on evidence for evolution. So are you pretty pumped about those debates or what?
Yeah. I'm just looking forward to the first one. I'll concentrate on the next one after that.
One at a time, right, brother?
One at a time. Yeah.
One at a time.
Evidence for evolution. That's an easy one.
Evidence for evolution. I mean, homology, right? We got those similar structures with all forms of life. These homologous patterns. What do you say to that, Matt? There's evolution right there. You're related to a worm.
Yeah. How come God made a whale, and how come he didn't make whale sharks? I mean, who knows?
Right. I mean, why do we as humans share any similarity with any other form of life in the biological world,
including other humans?
Right. Yeah. Believe it or not. So, okay. Well, I'm looking forward to that. That's going to be good. Let me go over a couple reminders for everybody as well. So the last couple of weeks, we finished a series of open mic discussions. I am now working on another series of open mic discussions. Yes, we'll be getting another one on the Trinity. We'll be doing another, at least 2 on eschatology. So we had a lively eschatology open mic after show in the Debate Dojo a few nights ago. That was fun. And we also did 2 on the Trinity, and I am talking to Team Hovind. We will be having another evolution open mic sometime in the next couple of weeks. So we've got events, 4 to 5 booked a week for basically the summer, and I'm even booking some big events for September. And so I'm excited, guys. This is going to be the summer of all debates. I knew it would be tough to top last year's summer of all debates. We had some epic, actually last year, all of last year was our Evolution Debate Challenge series. And I think we did about 60 or 65 of them. So for those that love a good old fashioned evolution debate, check the playlist section titled something like the Evolution Debate Challenge Series 2022. And there's endless fun there. Okay, so tomorrow night, for you guys in the States, it is a holiday, isn't that right, Matt? Memorial Day weekend, if I'm not mistaken.
Yeah, that's right.
So we got a Memorial Day debate. This will be Pastor Matt first, Charles Jennings. I love both these gentlemen, both great debaters, highly knowledgeable, sound on the bigger picture stuff like soteriology, deity of Christ, so on and so forth. But they're going to be entering the debate octagon. They're going to be debating replacement theology. So controversial topic. Definitely a lot of varying opinions on this one. So Memorial Day or night debate tomorrow night should kick off, I believe, at 9 EST. Next week, we've got quite a few more shows for you as well. So just real quick, couple announcements. This will be another soteriology debate. I know people are enjoying these. Is free grace theology biblical? Actually, we've got a replacement for this one. Josh has notified me he's got a few things going on, but Charles Jennings, keeping him as busy as I keep Matt, he'll be filling in. So Charles and Lucas Curcio, they will be debating this topic June 2nd. So the second June starts, we've got plenty of shows for everybody to enjoy. Mark White, my brother, I see in the chat. So I'll give your debate a quick shout out. Lots of people looking forward to this one. Pastor Scott Clem, Mark White, is there a geopolitical future for national Israel? And so, again, with that, I think we are going to, looks like we got a super chat that came in. Ian Chen Official, appreciate the support. Thank you for the super chat. He says, hi, Donnie, good to see Dr. Joel and Dr. Zach. So we got two of our biggest fans here in the chat. And Ian says, Raw Matt, when you're done, I'd love to debate you also. Maybe Donnie can set it up. Well, there we go, Matt. You're going to be extra busy in the summer. Everybody wants a chance at you. So, okay. With that, I think we're just going to get right into it with the formalities out of the way. I do want to say to anybody who is interested in debating whatever topic it is, shoot me an email, standingfortruthministries@gmail.com. Let me know what your 5 or 6, I guess, most preferred topics are to debate, whether it's soteriology, nature of God, creation, evolution, ancestry, you name it. And then that'll help me set you up with a worthwhile interlocutor. Okay, so I do see questions already coming in. I'll star those for later as we do want to get into the program for tonight. So Matt, I think we'll just start on the human-chimpanzee DNA similarity. So let me share screen. I got to make sure the audio is checked. We're going to play through one video here from Dr. Marcus Ross. So he's a fellow young earth creationist, fellow Christian. And the arguments he makes in this short clip are very similar to the arguments made by people like Gutzon Gibbon, Dr. Dan Stern Cardinal, so on and so forth. I believe these are honest errors, honest mistakes. And so we're just here to benefit our own camp, our own young earth creationist community, and basically point out where we think he goes wrong, but also where he may have a valid point. And so I think at least one question he asked is a legit question that I'd be interested in getting an answer to. So, okay, I got the audio checked. Let's go ahead and have some fun.
Well, Tompkins' approach is to look at every single difference amongst the human and chimpanzee genome. And the human and chimpanzee genomes are not exactly the same size. So it's not like you've got a 1-to-1 correlation all the way around. Sooner or later, you got more over here, or on some chromosome, you got a lot more over here. And he counts all of those differences equally. And that's how he comes up with a similarity of only 80%. So for example, let's say that, I've got this piece of paper over here that we put in some of the kits that we make at Cornerstone for curriculum. If I accidentally put in 5 of these into one kit, I just grabbed a bunch and threw them in, and the other kit only had 1, Tompkins' approach would recognize that as 4 differences. 2 that were the same, and then the 4 duplicates that shouldn't have been there. That's 4 differences. And so he does the same sort of thing with the genome. Anytime there's any sort of difference, a duplication, a deletion, or what have you, any of these differences at all are all counted equally. Well, that's a little weird because sometimes in our own genome, we have these long repeating segments that just look like something was copy pasted, copy pasted, copy pasted. Should you count that as 15 or 1? That's actually an interesting question. You could go either way, but when you decide to go one way or the other, there are going to be other kinds of consequences. So for example, right, I think Answers in Genesis would like us to be able to say that human beings are all within like 99% equal to one another. But if you take Tompkins' approach, you might not be able to say that either. Tompkins has never taken 2 human genomes and compared them and come up with a number of similarity, which might be something like 94%. Because if we're only 80% similar to a chimp, we might only be 6% similar to each other. That might not be a number, though, that Answers in Genesis or ICR likes to publish, because they would rather us say that we're all almost exactly the same. We're all human beings. But Tompkins hasn't given us a number for human-to-human comparisons. He's only given us this brute force difference between chimps and humans. How other researchers get the 99% or 98% up their way in the 90s is they are comparing the sections of the genes of DNA that code for proteins, that are coding for things that are being made in the cell. And when they see differences there, they find that there's very, very few differences between chimpanzee DNA and human DNA. So when you look at it on that level, you get 99, 98% similarity. If you look at it on what's the total number of differences, well, then you get like 80%. But you've got to make sure that you're comparing apples to apples and not apples to boulders here. Just because they're both round doesn't mean that they're fruit, right? So Tompkins, choosing the 80% for this article has not solved their problem because they don't have the human-human comparison. And I think that's important. The article is going after Todd Wood.
Okay, I'm going to stop it there. Matt, let me know if my audio is coming in okay.
Sounds good.
Okay, good. So I left from our second channel here. I left a comment in the comment section of this video. I also made a post, a lengthy post over on this channel, Young Earth Creation. So if you're interested, go check it out. A little more detailed than the comment that I left on the channel. And so you can read through that. I'll touch on a number of these points in just a second with some slides and presentation I put together. And the host here, whose name is Zach, I believe, he said, thanks for mentioning this. I brought it up to Dr. Ross and he said, interesting. Certainly I could be wrong in how I understood Dr. Tompkins' work. And so he might be willing to acknowledge some errors there. But with that, let me, I'm going to go over a few points. I'm going to go over it's a technical issue and it's an interesting one. It's an important one. And so when either side, I don't think all the answers are clear cut, but I want to give some of my thoughts. First, I want to start backwards with what he said on the nature of the similarity and how we come up with the percentage similarity. He basically pointed out, it kind of sounded like an evolutionist argument because he tried to say, well, the protein coding genes, when you just compare those, you get a higher similarity. And he almost implied that that's how we should do it. But that methodology, that starting point seems to be an evolutionist starting point because they're the ones that basically think the majority of non-protein coding regions in the genome are junk or evolutionary leftovers. That goes for endogenous retroviruses, pseudogenes, gene deserts, so on and so forth, right? These ancient repetitive elements, as I believe somebody like Dr. Francis Collins would call it. Well, firstly, I want to make a couple points on that. And that is, we now understand that there are many more long non-coding RNA genes than there are protein coding genes. And these long non-coding RNA genes, as you can see here, at least on this slide, are essential for healthy cell processes. But when it comes to the protein coding genes, they actually turn out to be, as you know, Matt, the building blocks, which is one of the many reasons why they tend to be more similar. They tend to be a lot more similar among creatures, okay? But the non-coding RNA genes, they're more taxonomically restricted or creature specific. And these genes actually provide the metadata for the building blocks that are the protein coding genes to properly function and carry out their various roles and jobs. When it comes to the long non-coding RNAs, they function in a variety of ways. Many of them function in the nucleus. They can modify DNA. And notice this paper here. Long non-coding RNAs are a diverse class of RNAs that engage in numerous biological processes across every branch of life. Gene expression patterns have implicated these long non-coding RNAs in diverse biological processes, including cell cycle regulation, immune surveillance, and embryonic stem cell pluripotency. And notice here at the bottom, there is a growing body of literacy. The days of this paradigm, one gene, one protein, one function, 40 years ago, that's what you would typically see in textbooks. That's been completely overturned. I mean, one single protein can have many different functions. It's called protein moonlighting. And so notice this, there's a growing body of literature from yeast to mammals suggesting the non-coding RNAs have an important role in chromatin state formation. Right here, I've got a whole PowerPoint presentation, as I have written a book on endogenous retroviruses, non-coding DNA. The functions of non-coding DNA include regulating expression of genes, maintaining genome integrity. So basically the process by which a gene gets turned on in a cell to make an RNA, to make RNA and proteins would be gene expression, okay? But then you've also got, so you got gene regulation, gene expression. And gene regulation is essentially the process used to control the timing, location, and amount in which the genes are actually expressed or turned on in the various cell tissues, okay? Or gene regulation is the process that basically turns the genes on and off. And we're going to focus on, Matt, in a little bit, where I think it's more clear cut. So rather than looking at the similar, are we going to look at all the non-coding regions of the genome and the protein coding or just the protein coding? Well, we can just look at things like gene expression, like gene regulation, and there's actually significant differences between humans and chimpanzees in terms of the way genes are expressed, in terms of the way genes are regulated. There's differences in alternative splicing. There's differences in genome architecture. There's differences, massive differences, as we know, in the Y chromosome, which I'm going to get into soon. Anyways, all of this is comprised in the non-coding areas of our genome, maintaining genome integrity, acting as mutational buffers, switching on and off genes. We've got work as regulatory elements during transcription and translation. So DNA transcribed and RNA translated into protein. So we got all these different functional responsibilities taking place in the non-coding DNA, not to mention we've got known functions for endogenous retroviruses, for pseudogenes. We've got documented functional roles for ALU sequences. Anyways, we've gone through this for hours and hours. So that's just kind of a brief overview. But my point of that, Matt, is since we basically have evidence for genome-wide functionality, okay, at least genome-wide activity, which includes all these sequences and DNA elements that evolutionists claim are junk or evolutionary leftovers or the ancient remnants of viral infections or just genetic mistakes. And even the areas in between genes contain many vital functions and act as control features for the genome, right? You got introns, which have known functions. And then obviously you've got exons, which are the protein coding sections of our genome. And so it's just function after function. And therefore, we should be in our comparison, correct me if I'm wrong, Matt, what are your thoughts on this? We should be considering your non-protein coding regions. We should be considering your protein coding regions, not just the protein coding regions. So yes, the areas of the genome that do not code for proteins, what we know based on the scientific literature, based on ENCODE, for example, is that they turn out to act largely in many of the species-specific, let me repeat that, species-specific aspects of DNA. And then many of your standard protein coding genes present in the genomes of biological organisms are performing basic biochemical functions. They operate as essential genetic building blocks, which is why it is these types of common code regions in the genomes that tend to be more similar between different types of creatures, right? Humans, chimpanzees, other primates, so on and so forth. But this is also why it's so important for us to compare the non-protein coding regions of genomes in order to get an accurate percentage in terms of how similar the genomes between humans and chimpanzees or humans and any other creature is. Matt, what are your thoughts on my thinking there?
I mean, it makes sense. You know what I mean? If we're looking in the entire genome, of course, there's going to be more distance and space between us in relation, right? That's why when we look at the mitochondria, there's a little bit more differences, and then we branch out and we look at the larger Y chromosome, and then we go, oh wow, we can see further back in the distance because there's more clarity, right? It's a larger aspect. That's what's so good about it for paternity testing. But they want to invoke that these other regions, like the protein coding regions, is the autosomal, right? They're trying to infer that that's the best way to do relations because they can't get a convergence by using mitochondria or the Y chromosome to converge on any missing link common ancestor. You see, that's the goal of evolution. It's trying to prove common ancestry, and they can't do that by looking at these regions that show paternity testing skills. So they like to invoke the hypothesis that, well, we can just look at this other region, but they don't understand that that other region is designed, it's created. That's what makes them different and unique, and how we structure and these protein coding genes function, what they're doing, what they're building. So yeah, I agree that we should be looking everywhere in the genome. Why not? Will it create more of a differences between humans? Probably, because we're looking at a huge number. We're looking at a massive amount. How many genes do people have? 20,000? 19,000? It's a lot. There's a lot of genes. So there's a lot to look in. There's a lot to look at. And the more you look at, the more differences you're going to find. But it also means the more differences you're going to find in everything else as well. So I'm fine with it. It doesn't matter. We'll figure out how close we all are when everything gets scanned. But they're not scanning everything because they don't think it has any function. Like you said, right? It's junk DNA. Why even look at it? It doesn't do anything. So we need to get that mindset away and then do comparisons. That would be great.
Well, especially as creationists, we don't want to come into this or into our arguments by invoking criticisms that seem to be based on an evolutionary starting point.
Right.
Like, for example, let me bounce a couple ideas off of you. I've been going through Erica's video and working on a separate response to that. It's a 2-hour video, and I've been going through it carefully, her video critique of Dr. Tompkins, and I'm about an hour and a half in. And so my question to you is, when it comes to weighting sequences or basically these gaps, okay, let's say we're looking at an indel, insertion deletion, right, when it comes to these mutations. And you'll see the evolutionists that say, okay, if we're looking at, a deletion here, let's say you're looking at a deletion of, 1,000 base pairs, hypothetically. Does that count as 1,000 base pair difference, or just one, a deletion, or even a duplication, right? Do we count this as one difference, basically a duplication mutation, or if it's duplicated, 500 base pairs, do we count it as 500 base pairs? But doesn't this all go back to the starting point of creation versus evolution, which is the assumption that this is a duplication, or the assumption that this is an insertion or a deletion? So if we see an area in the chimp genome, for example, Matt, speak to this a little bit. And so we see, right, so Zach Hancock, I appreciate that clarification. So let's just say one deletion event. And so we have in the human genome, right, we have a specific genetic region but we find a gap when we're comparing human and chimp genomes. And so this genetic region is not present in the chimp genome. Is it not an evolutionist assumption to say, well, this used to be there, but now it's been deleted through this mutational event at some point since the split, since the divergence?
Right.
Isn't there assumptions going into this in the first place?
Of course there is. They always have. Remember, their starting hypothesis is that evolution and then divergence and then more branching off, more branching patterns, and therefore over a deep amount of time, if there's discrepancies and problems, sure, deep time erased it. It scrambled it more. But then again, when it's not as scrambled as they expected it to be, they invented the bottleneck to say, uh-oh, that's why there is more similarity than we expected. Because if there were hundreds of millions or even billions of years, there would be nothing left. It would be scrambled beyond oblivion. That was their original prediction in the 60s, that everything would have no genetic similarity. So you and a crocodile could be tested, or you and a banana, there'll be zero genetic similarity. So what they did is they invented the bottleneck. The bottleneck saved them. And then they came along and said, well, sure, there's some discrepancies in how close we are similarity, but that's because things were deleted along the way, like you're saying. That's the problem, that the person whose video you played has the evolutionary mindset coming into this.
Right, because if we're going to say, okay, we've got one gene here represented as a paper, a paper in the human genome, but we've got 4 of that piece of paper in the chimp genome. Okay, so we should really only count 1, since we've basically got a duplication that made 4 in one lineage. But aren't we assuming, and again, speak to this, I'm just kind of brainstorming, just, throwing ideas off you. These are the types of questions I would ask somebody like Dr. Ross or Gutsy Gibbon. How do we know this is an insertion? How do we know this is if we're comparing humans and chimpanzees, creationists are starting with the assumption that humans and chimpanzees are not related and don't go back to a common ancestor. And we are building a model based on that. The evolutionist is assuming that humans and chimpanzees do go back to a common ancestor between 6 and 10 million years ago. And so they will assume that that is an example of a deletion event or an insertion event or a duplication event. But as a creationist, how do we approach that? Because are we also assuming that that is a duplication event or that is a deletion event since the split?
We could say that the best thing to do would be look at what are the percentages of gene duplications and deletions that actually occur. How often do we see them? How many events of these actually are happening? And then can we extrapolate that back in time to say that that's true? A lot of times these events don't occur as often as they think they do. So they invoke them constantly, but they're not a constant event that's replicated and seen very much. So that's one of the best areas to look. We've talked about that in the past. And
right.
Go ahead.
It's just like taxonomically restricted genes, like these orphan genes, right? We see these functional genes that are species specific oftentimes, and so we see them in humans, we see them in chimpanzees, but there's no evidence for their precursors, right? You see them, they suddenly pop up.
Right.
And it's the evolutionists that would assume, well, okay, we've got an example of de novo gene synthesis where a non-protein coding region of the genome, through mutations or whatever means or mechanism, became functional, which resulted in an orphan gene. Or, there's a duplicated region that was at first not functional, but then it was hit with a series of beneficial mutations and became a functional gene. And so as a creationist, we can say, well, there's no evidence for the precursors to this orphan gene because It is there by design. This is a functional DNA unit that is there due to creation. It's the evolutionist that has to explain why that's there. And to me, it's circular reasoning because, de novo gene synthesis exists. And it's like, well, why? Well, because we have these orphan genes. Well, why do we have these orphan genes? Well, because of de novo gene synthesis, right? So it's this big circle of them trying to explain it. But I don't want to go on our classic rabbit trail. So let me go through a couple points here. And then I want to play Dr. Tompkins' response to what Dr. Ross was saying here. Okay, so notice on the slide in the YouTube video, Answers in Genesis, that's the one that we just looked at, argues against the work of Dr. Jeff Tompkins. Dr. Ross's arguments can be viewed starting at that timestamp, which we did. Dr. Tompkins addresses these criticisms made by Dr. Marcus Ross at timestamp. So I've got a video here, Monkey Business in the Chimp Genome by Dr. Jeff Tompkins. I was actually originally scheduled to host this program as I'm doing work on endogenous retroviruses for Logos RA. And I had something scheduled that day. I didn't mind rescheduling it, but Chris Roop also kind of wanted to get in there and host a show. He's got a cool new studio and setup now. And so we decided, you know what, have Chris run the show. It turned out to be a great show, had a lot of critics in there, a lot of great questions. I thought the presentation was good. And so Jeff did respond to this specific criticism by Marcus Ross, which I'd like to play in 1 second. But first, before I do, I want to give my own thoughts. Okay, let me put it full screen so people can see a little bit better. Okay, so firstly, evolutionary biologist Richard Boggs, who Erica does speak about in her recent video, he acquired independently, mind you, nearly identical results using the latest version of the chimp genome. Okay, and a different algorithm. This strongly confirms the findings of Dr. Tompkins. Now, if Jeff included, see, here's where I think a lot of the critics go wrong, is they think that Jeff is coming up with basically a maximum, where he's actually coming up with a minimum. And why is because if Jeff included unalignable parts, such as large duplications, that seemed to be the illustration Dr. Ross was pointing to, was a duplication event, right? So he has, if Jeff included unalignable parts such as large duplications, the genomes would be even more different, probably 80%. And so he's actually being generous to the evolutionists, right? He's actually, for sake of argument, saying, okay, maybe these unalignable regions or these gaps, you know, let's not include them for sake of argument and see what percentage similarity we get. His estimate was based on alignable regions. All unaligned regions, including large indels, we talked about that earlier, insertions, deletions, were excluded from his percentage estimate. And this was all reviewed by Dr. Tompkins, actually. And so, yeah, he said this is correct. This is basically an accurate summary of what he did. In addition, these alignable regions included only small insertions and deletions, which were allowed by the liberal gap extension set in the BLAST algorithm parameters. This means that his analysis was merely an upper limit, so the actual percent similarity must be lower than 84%, probably closer to 80%, once unalignable regions and genome size differences are considered, as Dr. Tompkins notes in his paper. Dr. Tompkins' methodology allowed for him to be particularly generous with the percentage similarity. And so his numbers were a minimum. This is my understanding. This was also confirmed by Dr. Tompkins. Okay. His numbers were a minimum and not a maximum. But a lot of the arguments I see from the critics seem to be implying that what he's looking at is a maximum rather than a minimum. Dr. Tompkins has performed sophisticated analyses of human and chimpanzee DNA similarity, and Dr. Ross, well, see, it could be, given the comment that I received under that video, it could be an honest error. You know, there is a lot of work associated with this. There's a lot of papers associated with it. And I think if you're not up to date and you might just be repeating an argument that you've heard from somewhere else, you're just, as a result, it's going to be an error, right? But there's another pattern that the critics fail to wrestle with. You don't even have to reference the work of Dr. Jeffrey Tompkins. And I think this is key. All you have to do is read the supplemental material found in the evolutionist's own raw data. Okay, read the paper, Initial Sequence of the Chimpanzee Genome in Comparison with the Human Genome. And you will find far more differences between human and chimpanzees than 99%. And this is found directly in the secular literature. The evolutionary community have been less than straightforward when reporting the numbers on human to chimp DNA similarity. It is a fact that this is a fact. I mean, everybody has to admit there's large chunks of DNA not found in chimpanzees that exist in humans, and large chunks of DNA not found in humans that exist in chimpanzees. Now, again, I'm going to point out what I pointed out at the beginning is I think the better way to argue is through the differentiating lines of evidence, like the Y chromosome dissimilarity. When you consider overall architecture, gene content, size differences, the Y chromosome, which is uniparentally inherited, non-recombining DNA, it should be relatively more stable, and yet it's only 35% the same. And there's no controversy with that number. I mean, this is a well-known documented fact that the Y chromosome is only 35% the same. And that is a huge issue for the evolutionary community, which I'm going to get into in a little bit more detail in a bit with some citations and excerpts. Also, the gene regulation differences, the way genes are expressed, alternative splicing, genome architecture, there's epigenetic differences. I think these are so basically, even in your similar sequences, right, Matt, between humans and chimpanzees, there's differences in the way those genes are expressed or regulated. And I think that's important to consider as well. Okay, the evolutionary community, again, they've been less than straightforward. Now, here's some questions. Firstly, Matt, speak to this a little bit. And the question that I don't necessarily have here is, what if we went with the 96% number between humans and chimpanzees? Is that a problem for the creation model? Let's say, rather than the 84% similarity, because regardless of if it's 84% or 96%, there still is a relative hierarchy that exists in the biological world. And what I mean by that is humans are more similar to chimpanzees and the other great apes, right? Gorillas, orangutans, bonobos, so on and so forth, than humans are to any other form of life. But banana plants and humans, they share 50% of DNA we share 50% of our DNA with banana plants. And so since we already share a lot in terms of anatomy, morphology, and physiology with the chimpanzee, is it really a surprise that we're going to share more with them overall? And I'm thinking genetics here than, let's say, with a banana plant or a whale. And so basically, yeah, my point is, speak to this a little bit, Matt. Is there an issue if we went with the higher number as compared to the lower number, and then decided to focus on differences involved in gene expression, gene regulation, epigenetics, alternative splicing, and also the Y chromosome? Go ahead, Matt.
It really wouldn't matter what the numbers were giving, because they would still eventually make a hierarchical pattern, because we're looking at specific regions only in the nuclear DNA, specifically a protein coding gene. They can build any tree that they want by looking at different areas. That's why when they look at cytochrome C, it builds a different tree than cytochrome B. So they cherry pick the data that they need to build these trees, but they don't use anything like a paternity testing to do it. Because like I said, when you look at the Y chromosome or the mitochondria, you don't go back to any convergence. That's what evolution needs to do. It's not there. So they look at the nuclear DNA. And we explain nuclear DNA as the created differences built in, because, like, something has to be constructed and built out of these proteins. There has to be some type of a function, and that function is building and creating organisms. And the more similar we look and function, the more similarity is going to be in there. It's not because we're related. It's because of that alone. Very simple. But that's the difference between the creation model and the evolutionary model. They're starting at different viewpoints and different starting points. We give an example, they give an example. Ours, we actually look and do a paternity test and go backwards in time, and we can literally test it. They can't test theirs. So they cherry pick certain aspects inside the genome and just go with that. Very, very simple. So it wouldn't matter if it was 95 or 80 or 85. Those numbers are irrelevant, because it's still going to build that hierarchical pattern while looking at the nuclear DNA. So he is gone. It's okay. I guess I can still go. What we got on here? Oh, he's back. There we go. I was going to start reading your list.
You just keep going, brother. You're giving some great points. So, firstly, when it comes to the hierarchical patterns, anyways, why would God not create some organisms that are more similar to other organisms, and also more distant to other organisms, right? By definition, correct me if I'm wrong, Matt, humans have to be more similar to some creature. So let's just say, so obviously humans are most similar to chimpanzees. But let's say, just take chimpanzees out of the equation. Take chimpanzees and bonobos out of the equation. Well, you're still going to have a hierarchical pattern in the biological world, because now humans are most similar to gorillas. Remove gorillas. Okay, humans are most similar to orangutans when you compare any other form of life.
Right. Or a pig.
Right. Take away all of your great apes. Humans are still going to be most similar to some kind of monkey. And so there's still going to be a hierarchy, because that means humans are going to be most similar to, let's say, a lemur than a human is to a dog or a pig or a fish. And so unless God created humans without DNA, RNA, protein, without, similar structures, similar bones, similar muscles, then obviously there's going to be hierarchical patterns in the biological world. I mean, was God supposed to use completely different building blocks, just alien building blocks for humans, and then something totally different for the rest? I mean, obviously humans got to fit somewhere in the biological world. They don't just fit over here and then here's everything else. That doesn't make any sense.
It doesn't. How could it? We couldn't even live. We wouldn't even be able to eat the food that's on the planet if we were so genetically diverse. Makes no sense. I mean,
right.
A matter of fact, imagine if we were so genetically diverse than everything else, we would actually believe that there were probably two creators then, one for the creator of animals and another God for the creator of mankind. We might actually believe that there would be multiple deities that created us if we didn't find the fingerprint of God on every designed creature on Earth.
Exactly. That's a great point. So the fact that we actually see the similarities, the fact that we are built with the same building blocks, points to one common creator rather than some polytheistic version where you've got multiple gods essentially. And also we'd have to be designed with these similarities and designed in these hierarchical patterns in order to advance in medicine, in order to use mice, for example, which are mammals and do have a lot of similarities with us. And so we can test various drugs and medicine. We can learn about the biological world. We can learn about ourselves. Genetic knockout tests are pretty unethical when it comes to humans. And so there is a benefit to having biological organisms existing on this planet that we share a lot to in order to advance in knowledge, in medicine, in healthcare, and so on and so forth. But if we didn't have those similarities, if there wasn't this hierarchical distribution within the biological world, then we couldn't advance in the ways that we do. We wouldn't have the knowledge that we have today. So, okay. So a question I would have then, and I think this is a valid question, is what happens then if we're going to go to utilize Dr. Tompkins' methodology, essentially, and go with the 84% number? What happens if we compare, and Dr. Ross pointed this out, and I think it's a valid question. What happens if we compare other organisms using the same methodology as Dr. Jeff Tompkins? For example, chimps to chimps. We understand there's a lot of diversity within chimpanzees.
Right.
And some chimpanzee populations are extremely different from each other. In humans, there is incredibly low genetic diversity, incredibly low genetic variation. That's you're talking about earlier. That's why they had to invoke the hypothetical population bottleneck, the hypothetical out of Africa near extinction event, in order to explain how high levels of heterozygosity could basically change to low levels of more levels of homozygosity, low genetic variation. And so we've talked about that in great detail. But so I guess my question would be this to you, Matt, because a lot of your critics, they'll say, well, you want to say humans and chimpanzees aren't related and you want to look to genetic similarities, but why are there some species within the same kind? Let's say felids or canids. Let's say felid. So if we as creationists say tigers, house cats, and lions all descend from the same archetype, that archetype front-loaded with pre-existing functional genetic diversity, where you got a lot of heterozygous genetic locations. From there, with the processes, recombination, gene conversion, isolation, inbreeding, breakaway population, so on and so forth, you get new species, right? All based on that pre-existing ability to vary and diversify in the first place. Okay, so let's say within the same kind, let's say felids, there's actually more diversity within the same kind, as in your, let's say your lions and your house cats, or your lions and your cheetahs have more DNA differences separating them than humans and chimpanzees. Yet we would say all those felid species belong to the same kind, but human and chimpanzees exist as separate kinds. What do we do with the DNA differences? How would you respond, Matt?
This is very easy, because if everything came through the bottleneck at about the same time, which is exactly what we see with the orangutan, the gorilla, the chimpanzee, and the human. So what happens? We're all coming through a bottleneck at exactly the same time. Cats as well. So cats come off, they start diversifying. Why would a chimpanzee have more diversity than a human being? And why would an orangutan have double the amount of diversity as a chimpanzee? If they all got off the ark at the same time, this is very easily answered when we look at generation times. Because if every single generation is accruing more mutations inside of them, then they're going to have more genetic diversity in each of these lineages. So when you look at cats and they have massive amounts of genetic diversity over a broader amount of time, it's very simple. It's because of their generation time. So the reason that chimpanzees have, they're more similar to us, it would simply be because when we're looking at the mutations of them, we have a 30-year generation time or higher. Chimpanzees have it 20 years or younger. And then we get into orangutan, theirs is about 10 years or 11 years old, right? So they've had twice the amount of mutations occurring in them than we do with them. So every time you look at that, it builds what looks like a hierarchical pattern. It looks like the orangutan has been alive twice as long as the chimpanzee. And it also looks like the chimpanzee has been alive longer than the human beings. Why? Because each generation is occurring more mutations, and those mutations make them look older when they're not. We all know that same bottleneck happened. The evolutionist just believes it happened 200,000 years ago, and the creationist believed it happened 4,500 or so years ago. So no matter what, there's still a genetic bottleneck, and all of those creatures went through it. And yet the genetic diversity that we see in all those different lineages is explained from the generation times that occurred after it. Very simple.
Now, Joel Duff in the chat, appreciate your comments, Joel. He says, and I'm looking for my phone here. He says, Matt, you are admitting that mutations add genetic diversity. Amazing. How would you respond to that?
Yeah, that's how we count. That's how we look at diversity. I guess we could also look at phenotypic diversity. And if we do that, that also validates my thing even more, because when we look at humans, there's massive amounts of phenotypic diversity, but the phenotypic diversity between the three major groups of people, the Caucasian, the Asian, and the African, the phenotypic diversity is exactly the same, which is what we would believe when Noah's grandsons inherited different regions of the earth, and the family spread out after Babel, inheriting those lands. Ham's line down in Africa, Shem's line over to Asia, and then Japheth's line up into the northern regions. But yet, that's exactly what we would see with phenotypic diversity, is it all is the same. But if humans originated in Africa, phenotypic diversity within Africans would be much higher, because they existed about 200,000, about 50,000 years before Asians and Caucasians even existed. So therefore, the phenotypic diversity would be in conjunction with mutation differences. So I like mutation rates just the same. If we're going to count them for diversity, it's okay with me. That we can do that game too. And that is, if that's one way to count differences, then so be it. But there's many other ways we can. And I like phenotypic diversity as well.
And what I find interesting about African diversity, how they look too, the fact that everything basically nests within Africa, and Africa has more diversity when compared to non-Africa. Basically, all you need is selective sweeps, or even one selective sweep, let's say across Eurasia, sometime after Babel, that resulted in your older lineages basically being wiped out with older lineages in Africa remaining. So your older lineages wiped out, older lineages in Africa remain or survive. And so Africa could have had a higher initial population size, Also where many people out of Babel moved into Africa, right? You got waves and waves in Africa. And so Noah's descendants are moving into Africa. And as a result, they're bringing in a lot of diversity. And so Africa is starting out with a lot of diversity, but the evolutionist wants to assume that this epicenter, because basically wherever you put or place the epicenter of genetic diversity, everything else is going to nest within that. It's a side effect of where you place it. And so whether it's a selective sweep or people are moving into Africa after the Babel event, Africa is starting off with high levels of genetic diversity. And so by definition, you're going to have that reflection. You're going to have that artifact, basically that nesting artifact, where the evolutionists want to interpret that as being because Africa's older. Goes back hundreds of thousands of years, no literal Adam and Eve, so on and so forth. What are your thoughts on that, Matt?
As far as what we would expect to see from a literal Adam and Eve aspect?
Well, basically with Africa, if the evolutionist wants to place the epicenter of genetic diversity in Africa,
okay,
from there as a side effect, just by placing it there, you're going to have that nesting that you see where everything nests within Africa. But it's not necessarily because Africa's older. It's because of the various things we've talked about, right? Selective sweeps, more people moving into Africa after Babel, where you have a higher initial population size, basically. And, Noah's descendants are moving into Africa, wave after wave after wave of people moving in, bringing in lots of diversity for Africa to start with.
Exactly. And then they got landlocked in there. So you got to remember they go into Africa, and then all of a sudden the Sahara Desert grew, whether it was from the icing ice off from the Ice Age. They got trapped. The Sahara Desert was there. Now they're living in Africa in small populations going everywhere. And you know what small populations do with mutations, right? It makes them reach fixation very quickly. So they're accruing more mutations because they have more recombination, and they have all these other aspects, PRDM9. So they have faster mutation rates in general. Then they have fast generation times, and they're in smaller population sizes. So literally, that's why they look older when that situation can arise in any population group at all. But what do we know about Africans? We know that they didn't have any farming or agriculture and never had a massive city that they built like in other parts of the world, like in Egypt and things like that. So what happened down in South Africa? Well, they stayed in little rural populations because you can't have a civilization when you're not growing grain. So they never grew that thing. So they can never accrue a massive population like other parts of the world were doing at that time. So it looks like they're much older.
Yeah, that's good. I'm looking at it. So there's some questions that came in. Slam RN. You know what? We'll take a 2-minute break in about a couple minutes once we finish with these questions, and I'll put the questions up on screen so we can kind of deal with. So, okay. A good question, I think Dr. Marcus Ross brought up, has to do with comparing humans to humans or chimps to chimps, wolves to domestic dogs, using the same methodology used to compare humans and chimps, the type Dr. Tompkins does, right? Because the question would be, without another reference, can we effectively utilize the DNA similarity argument from Dr. Tompkins? Gutsy Gibbon pointed that out. Also, what happens when we incorporate more species and compare the results to the human/chimpanzee results? And something we just discussed, why do some species within the same kind, say felids, for example, frequently have more genetic differences separating them than humans and chimpanzees? I think another reason, too, is because, remember, God created how many human beings? 2, Adam and Eve. And so what would we expect? We'd expect low genetic variation because that would restrict genetic diversity. You're starting with 2. With animals, a lot of people get confused. They think, oh, God created, 2 kinds of dogs, 2 felids, 2 elephants, 2 this, 2 that. Well, yeah, 2 of every kind went onto the ark, 7 of some. But at the creation event, God created an earth fully formed with fully working ecosystems and populations of animals. And so right off the bat, you'd have a lot of genes floating around these populations that aren't just comprising 2 individuals like humans, right, Matt? You have populations of the original dog kind, whatever that looked like. Okay.
Right.
And so, because as time goes on, as we talk about, you get these shifts from heterozygosity to homozygosity. You're losing a lot of genetic variation. You're losing orphan genes. You're losing a lot. A species will hit walls. So the further you go back in time to the arc 4,500 years ago and to the creation event 6,200 years ago, you'd have more levels of heterozygosity, less deleterious mutations. One thing I'd like to say to what Joel Duff was saying is, yes, a mutation adds something because it adds diversity, because it's adding something that was not previously there. But the question is it beneficial diversity? And I think the answer to that is mostly no, with the rare exceptions, of course. And it's the kind of diversity you get on your car. You get a brand new car and you get it off the lot and you drive to the grocery store and you park too close to somebody and, their door hits your car, leaves a ding, right? Your dings, your dents, your scratches, your rust marks. Yeah, the car is becoming more diverse. Or copy out the same book 100 times, introducing typographical errors. Yeah, the book is now more diverse, but it's not better. The message is lost. There's more mistakes, more typos. So yeah, you can say that a species is more diverse because of mutations, but doesn't mean that it's better. Just like your car after 10 years is more diverse because it's got a repertoire of dings, dents, rust spots. And so the evolutionists would have to prove that the majority of these mutations are providing useful or beneficial diversity. What are your thoughts on that, Matt?
I mean, I don't really see anything that's coming good from mutations. I don't know of anybody I've ever asked if you wanted a mutation right now, would you take it? They would all say no. So in their imagination, in their fairy tale belief system, sure, they can say that they're good all they want. Science says otherwise. Medicine says otherwise. They can believe that they're good all they want. Sure, you can count them for diversity all day long, but they don't want them. You don't want a somatic mutation and you don't want a germline mutation. They cause problems. And the more of them that add up, the worse things are going to get. That's why they say right now, like, oh, sure, yeah, evolution, the main parts of it have already happened. We're on the tips of the nodes of all the branches. So we don't expect to see evolution like we used to in that linear progression. Oh, really? That's what your theory is still saying. So they're basically saying evolution has stopped. We're only at the end of the branches and we're only seeing diversity spreading out now, right? So they're basically admitting what the young earth creationists are saying, exactly what we should be seeing. So we don't have to invoke anything. It's their stories that are made up that they have to now say, oh, well, yeah, we could just go back. We can invoke that. But that's their imagination of mutations are quite astounding, really. They put a lot of hope and faith into them because that's how they explain everything. Remember, it's beneficial mutations that drove evolution. Our mechanisms are entirely different. They don't know the biblical model, so I don't blame them for having complete ignorance on the subject, but at least learn about what the subject is before you ignore it. Kind of like Joel Duff. I remember watching his video on genetic entropy. Did you know he thinks genetic entropy is an animal getting smaller? He doesn't even know what it is. He doesn't even know line 1. He didn't even Google it, I guess. Oh well.
Joel, Matt's coming at you. Joel's in the chat. You're coming at him. That's good. Bring in the heat. So good points. And so basically then I believe, going back to this original question, why do some species within the same kind frequently have more genetic differences separating them than humans and chimpanzees? It appears that God created animals with a greater ability to diversify and adapt to environments in the first place. Because again, animals are created in populations. That's what the Bible says. But that's also what the genetic diversity in animals tells us. You get the rare exception where you have an animal species that's less diverse than humans. But overall, humans are one of the least diverse species, if you want to call it that, on the planet. Incredibly low genetic variation in humans. Again, chimpanzees, lots of genetic diversity floating around chimpanzee populations. So the Bible says God created two people, Adam and Eve. Right off the bat, that restricts genetic diversity. What do we see today? low diversity. If evolution were true and humans went back a couple 100,000 years to Africa, and before that to Homo erectus that lived on the globe for 1,000,000+ years, before that habilis, before that these Australopithecine-like ancestors, and every single generation mutations are being added to the genome, that would be what? That would be a lot of genes floating around these populations. And so today we should expect humans to have high levels of diversity. But when they discovered humans have low levels of genetic variation, they went, what in the world? We didn't predict this. And then that's where they invoked, which I would say is post hoc ad hoc, the out of Africa population bottleneck. And so it appears that God created animal kinds with a greater ability to diversify. And so, yeah, we as humans have low genetic variation. And even if we share 96% with the chimpanzee, there could still be separate ancestry there, because you're not necessarily comparing apples to apples, because now we're looking at animals when it comes to chimpanzees. Or if you're looking at the felids, so for example, your tigers, your lions, your house cats, your leopards, your cheetahs, your jaguars. Okay, yeah, maybe some of those species that go back to the same kind, or maybe there's multiple created kinds there, they may have, in certain instances, more DNA differences separating them than humans and chimps. But again, if we're looking at animals, we have to understand that when you go back to the creation event, God created animals in bulk. There's a good word. God created animals in bulk. With humans, he created two. And I think a prediction we can make involves genetic diversity. And it appears that it's consistent with that. And so I think that's one reason why some species within the same kind, felids, for example, frequently have more genetic differences separating them than humans and chimpanzees. And of course, the reasons you talked about as well, Matt. And so what are your thoughts? If they have this major issue with Dr. Tompkins and his methodology, if they were to compare humans using the same methodology, would we get the 99.9% similarity? What if we got something like 90% or 93%? Then would you say it's probably a safe bet for us to stick with the 96% number? Because as we've pointed out, the 96% number really is not a problem.
Yeah, it wouldn't matter either way. We would just go with the newest updated information and go with that. It would be nice to know the entire genome, right? If we're gonna be counting orphan genes, some of them are very small. The smaller orphan genes there's gonna be a lot of them. So we're going to have to count. Does that mean that every person has the same orphan gene? No. Remember, one of the predictions would be that Africans would have less orphan genes because they had more generations of younger reproduction. And that's what we found in flies as well. So if we find Africans have lower amount of orphan genes, then it would validate Dr. Nathaniel Jensen's predictions that Africans had generations of 15-year-olds basically having children and offspring to account for his mutation rate. So therefore, yes, we would see some differences, of course, because again, we're looking at a broader picture. Instead of looking at a puzzle piece with 100 puzzles, we're looking at a pieces that have 1,000, right? A puzzle that has 1,000 pieces. Well, the more you get, the more there's going to be differences. It's okay. Now all of a sudden you're looking at a genome with 20,000 genes. Yes, there's going to be more differences, but it doesn't matter. Like I said, we're going to be comparing things that are going to still be making a hierarchical pattern, right? So There's just going to be bigger differences between everything rather than smaller. It's okay. The more we want to know more, we don't want to know less.
Right. And, I'd be interested. See, here's my recommendation, some constructive criticism, I guess, right, Matt? And you can speak to this too. I'd like to see your thoughts. With everything going on, especially on YouTube and, going back and forth with the critics, I think it would be beneficial, especially when it comes to Dr. Tompkins' methodology, since there's a lot of attacks right now on his methodology specifically. He's going to know the details on that. He did the work, right?
Right.
And so where we see somebody like Gutsy Gibbon putting out a video where a lot of it deals with, like, for example, everything here, this is my thoughts on it. It's been confirmed by Dr. Tompkins, but some of the details where Erica talks about humans to humans and how different they are genetically when using Dr. Tompkins' methodology, it would be beneficial, I think, if somebody like Jeff utilized YouTube, utilize what we utilize, where we can do these response videos and we can go point by point, where he can basically just play Erica's video, allow her criticisms to come through, and he can respond. And we need that for the body of Christ. We need that for the world of young earth creation, because it's helpful to people like us that are young earth creation apologists to see how somebody like Jeff would respond to some of these criticisms, dealing with his methodology. What is your opinion on that? Do you find that a lot of the young earth creationists are spending enough time with the critics, or do you think they need to spend more time with the critics? What's your opinion on that?
They need to spend a lot more time, because basically the only people that are really doing it actually is you and me. Every so often they'll make a post, they'll write something, right? Like when anybody attacks genetic entropy, Carter might get together with Sanford and they'll write an article on CMI, but they're not going to hop on camera and come on here and say, okay, who made a video against me? Let's take that apart. They are either obligated by the company they work with that no, you can't do that, or they're like, that's not Christian. Let's not be mean to them. Let them say whatever they want to say. Whatever the rationale is, we don't know what it is, but they have one nonetheless. Right. So it ends up just being us. And if they don't share that information with us, we can't even defend them. So oftentimes we send them emails and we directly try to contact them, but a lot of times we're out of luck, because this is their work and we're actually willing to defend it as long as we know that you'll respond to us and give us what you say we should answer. Yeah. Otherwise, we're going to give our answer and they might not like it.
Well, I asked Dr. Jeff Tompkins long time ago, months and months and months ago. I asked him what his thoughts were on Erica's video and her claims that the chromosome 2 area was the only spot in the entire genome where you find the telomeric calling cards in both forwards and in reverse, right? TTAGGG, CCCTAA. And I asked what his thoughts were on that, and if that's what he discovered as well, and what's a good response to that, and so on and so forth. And he never got back to me. I know other people emailed him as well. He never got back to them. And so just constructive criticism, I think some of these PhD creationists, I do think they need to be more attentive to the critics, know what the critics are saying, responding more, helping other young earth creationists that are inquiring, because these videos are out there. And, Matt, we've written our own books, right? Here's 2 that I've written, and I can defend my own work. And I do debates, response videos. Like, we don't let these critics just get away with criticizing us without getting a response. So anyways, long story short, praise the Lord, me and Chris Roop, we looked into it ourselves. And so we didn't get a response from Jeff. So we looked into it ourselves, and I put out an article, several videos on it, so on and so forth. And we found that Erica's claim or challenge there was wrong, that the chromosome 2 was not the only spot in the genome that we find the telomeric calling cards in both forwards and in reverse in a head-to-head organization. And so I've documented that, and we did that work ourselves and basically solved that challenge. And so the point is, and it sounds like you agree with me, I think on some of these issues there needs to be more attention directed at some of these critics.
It would be,
and nice. I would say Erica's latest video, there's some points she's made that are directed to Dr. Tompkins and his methodology that only he's going to be able to answer. I mean, how are we going to be able to get in his head and know how to respond, right?
Exactly. I mean, at the same time, that is kind of our job now. We've kind of taken that role, and I think they like it, because it's exhausting debating crazy. How does it feel when you open your email and you have 50 unread emails, and most of them are just like, trying to get on your channel and give their point of view, and then tell you you're wrong, and then threaten your life. I mean, there's always something every single day. You're like, it adds up, right? So there's a reason they don't like doing this. It's nice being able to probably sit in the ARC encounter in AIG and just kind of like chill out and write, and then set up one debate in 2 years from now. That would be pretty cool, but we don't do that.
No, I mean, we're here to take on the critics, their arguments. So I was going to respond to something Joel said. Oh, he said, no, Erica didn't make that claim. She was just talking about the density. No, she included the density. And if you watched the video that you and I did, right, Matt?
Yeah, you played the clip.
Right. I played the clip where she literally, and I rewound it a few times. She said, it is the only place in the entire genome where we find the telomere calling cards in both forwards and in reverse. And then later on, she said, and in that density. So the density is part of the argument, but she also had, as part of the argument, the head-to-head organization. And so, but I mean, we've addressed all that. We're beating a dead horse with that. There's been no response or rebuttal to those points yet. Anyways, okay. So I wanted to play Dr. Tompkins' brief response to Marcus Ross on this issue. And so let me play that now. Let me know if the audio comes through. I'm going to hit mute so there's no echo. And here we go.
Create these maps where you can connect genes, based on their expression into networks and assign functionality to them,
or you can,
based on the processes that they're associated with, the tissues that they're expressed in. I discussed this in my papers associated with that particular gene.
Very good. Thank you. So this is our final anonymous question. And then after that, we're going to close. So this is, here's the question. A well-known creation scientist, Dr. Marcus Ross, has stated in a recent interview on YouTube that your estimate of 80% or 84% chimp human DNA identity is inaccurate and should be closer to 94% because your analysis counted all differences equally, including duplications and insertions, and does not account for genome size differences. Is it true that if you account for these things, your estimate would be closer to 94%?
No, actually, if we included the regions that were dissimilar between human and chimp, and we included the dissimilarities in genome size, which they've actually measured with flow cytometry, that it would actually be lower than 84%. And so Marcus Ross is a paleontologist, and I don't know where he got his information about my research, but I actually did listen to what he said, and I hate to say it, but he misrepresented my research and did not describe it accurately, really at all. And so if Marcus Ross wants to give me a phone call, I can get him on track with what I really did and what the data really said.
Okay, and we have one more question that was.
Okay, so this goes back to what I was saying in my slides, that Dr. Tompkins' numbers were a minimum and not a maximum. And so he's pointing out that a lot of the critics, or Dr. Marcus Ross in his video there, was making it sound like these incredibly dissimilar areas Tompkins was including, but he actually was not including those. And that's my understanding too, looking over his work. So if Jeff included unalignable parts, such as these large duplications, the genomes would be even more different. And so he points out that his estimate was based on alignable regions. All unaligned regions, including large indels, were excluded from his percentage estimate. And so I don't know what the critics in the chat think about that. I guess we can see, but okay, we'll see what the critics say. But anyways, so if his estimate was based on alignable regions, and the alignable regions included only small insertions and deletions, which were allowed by the liberal gap extension, then that means his analysis was merely an upper limit, right? Which means it could be even less similar. Now, this goes back to what we were saying, Matt. The word duplication, the word insertion, the word deletion, these mutations, evolutionists assume that all DNA differences within species, across species, are the result of mutations over time. But we as creationists, we explain the vast majority of DNA differences, especially in autosomal DNA, as being the result of created nuclear heterozygosity. And so if humans and chimpanzees aren't related, but we see a section in the chimp genome that's not there, but it's present in humans, the evolutionists would assume, well, this is due to a deletion. Or maybe we have 3 of a certain gene as compared to 1 in humans, they're going to assume it's a duplication. The question is it a duplication? Is it a deletion? Is it an insertion? And that'll help us answer the question, how do we weigh these sequences? Does that make sense what I'm saying, Matt?
Yeah, I mean, if they're, like you said a little bit earlier, if they're not counting things that are missing, then obviously by counting those things, the gap is going to widen even more. I like that. So simple.
So, no, I appreciate that. So Joel Duff says, sure, it would be more different. So, Joel, would you be agreeing with Dr. Tompkins that Marcus Ross may have misrepresented his work a little bit? And then he says, kind of like if you compare. So basically he's saying, so if he compared me and you, Matt, in the same way that Dr. Tompkins compares humans and chimps, we would come out as 90% similar rather than 99.
Well, not that high. It wouldn't be down to 90%. If chimps are only 80, we wouldn't drop.
Well, he's basically right. saying that these critics are criticizing Jeff Tompkins' methodology and saying his methodology, when used on other species or even internally, like within humans, you're going to get a lot less difference. But they're basically saying it's inconsistent.
I see what you mean. No, we definitely might get lower because, again, when you're looking at more pieces of a puzzle, you're going to find more differences. So obviously looking at the entire genome, there's going to be more differences that we find between us. But then again, it's just going to broaden that gap looking at chimps as well. So if they're at 80 and then we start counting humans and we drop to 95 instead of 98, then the chimp's going to drop to 75. It's just going to largen the gap. The more we learn, the more understanding there is. We'll have that genetic boundary still exists. So it doesn't really matter. The genetic boundary, they'll never be able to overcome it. Just like with the taxonomic boundary paradox, right? They can't overcome it. They have to invent their own. Same thing with this. The genetic boundaries that show the mutations converging on a non-existent common ancestor of anything other than a human, just like nothing but a chimpanzee for a chimpanzee, they can't get around that. So it doesn't matter what that gap really is. It'll just draw the line of difference between us even more. It's good.
So let me ask Joel then, if humans and chimpanzees using Dr. Tompkins' methodology brings us to 84%, and let's also say we include all of your unaligned, all of your like gaps, large duplication, so on and so forth, these unalignable parts, let's say it brings it down to 78 to 80%. Is Joel saying that humans would be 90%? Because that would still be a 10 to 12% difference. But then they're going to say the gap shortens because, well, 12% difference between humans and chimps and a 10% difference within humans. Well, we would agree all humans are related. So how do we explain that 10% difference? So it's an interesting question, but it's also why it goes back to, okay, so Joel Dev says, okay, let's grant Matt's point. So a wolf and a fox are 70% similar using that method. So you agree that there is a boundary between them. Well, that depends. Are foxes and wolves related through common ancestry, Matt? With your predictions, what have you discovered? Do you think foxes fit within the greater canid kind?
It's either fox or coyote. I know the coyote is. I'm pretty sure I'm confusing the two, but I know coyote is related to the basically the dogs that trace back to the wolf. So I'm pretty sure that it would probably fall within that same kind. Again, there's a generational pattern, so there's a much more diversity in them because they don't have just one or two childs. They have a litter. So when they have a massive amount more diversity going on, because a human doesn't have a litter of human beings every time they have one decides to have a family. So whereas animals, they're diversifying, they're migrating, and they're speciating, and they're having massive amounts of puppies, and that we expect a lot more diversity based on that aspect. So that you kind of got to look basically and trace the mutations from that Y chromosome, go back, where does it land? That'll be your common ancestor right there. And
I'd be interested to see because remember, this goes back to what we were saying earlier. If we're going to use one specific methodology that makes chimps and humans 84% similar. We have to be consistent and use that same methodology for other kinds. We have to use that methodology for humans, but that might make those kinds and us as humans more dissimilar, right? So rather than us being 99% similar, 99.999, then maybe within humans, there's examples of 90% similarity. And so then if that's the case, the question is, what are we going to go with? Humans are all 99.999% similar, but then humans and chimps are 96% similar? Or would we go with, within humans, you can get 90% similarity, but then chimps and humans, 84%. You get what I'm saying? So it's like, it's one of those things where I'd like to see how Dr. Tompkins responds to these criticisms of his methodology, because it would be nice if we could have humans, right, Matt, speak to this. If we could have humans at 99.999% similarity, like we do, and also have humans and chimps at 84%, that would be ideal. That would be detrimental to the evolutionary story, because that's a lot of DNA difference. That's about 300 to 400 million DNA differences they have to account for. But that is going to depend, I guess, on the effectiveness of his methodology, because we were talking about earlier how the 96% doesn't pose us an issue, because regardless, you're going to have a hierarchical problem or a hierarchical reality, and other differentiating lines of evidence that we'd focus on, that I think I'm going to focus on in a minute here. So I guess as we wrap this section up, these are my thoughts. Okay. These are my criticisms, but these are my questions, these are my curious questions. And I think Dr. Marcus Ross asks a good question, credit where credit is due, on the human to human DNA comparison, or within the canid kind, domestic dogs with wolves with coyotes. Let's see how many DNA differences separate them. But it's also going to depend on how well we have sequenced those genomes. Isn't that right, Matt? You know, with various wolf species or felid species, do we have good DNA data to even
on those? We've sequenced worms and fruit flies pretty good. We have their entire genomes mapped out for scientific reasons, right? So we can map a nematode and things like that going all the way back. So there's some genomes that we completely have. That's why Dr. Nathaniel Jensen was able to get de novo mutation rates on those things and make predictions off of them, right? Because those have been mapped out really good. As where like a random species of alligator, probably not. Chicken, maybe so. That would be even more handy dandy. But I agree. I think the more you specifically broaden your branch of what you're looking at, you're going to get more differences. Let's just take you and your wife. If they ran your genomes and then said, how much of a difference is there between? Well, guess what? Your wife doesn't have a Y chromosome. So she is instantly 58 million base pairs different than you. So by looking at the entire genome, of course, there's going to be massive differences. There's differences between everything, right? So it's not about how big the differences are. That's more of like a Tompkins trying to push apes even further away. I just like looking at the mutations, trace them back, and where do they converge? Very simple. We know what came out of the bottleneck because genetic diversity is reset at a bottleneck. Follow the mutations back. Do we converge on any common ancestor? Does a bear and a dog converge on a bear dog? No, it doesn't. End of story. You know what I mean? They can play with their fossils all they want. That's why we have bearmanology. They can play with the fossils. Let's look at the genetics. The genetics doesn't say evolution is true.
Right. And this is basically what brings me to this next section. And then after this section, we're going to get your thoughts on this video attacking mitochondrial Eve and Y chromosome Noah. Okay, so the human Y chromosome. There's no controversy on this. Here's at least a picture where people can see just how different the human Y is to the chimpanzee Y. And as a matter of fact, there's a break in the hierarchy within the Y chromosomes because the human Y is more similar to the gorilla Y. than either are to the chimpanzee Y. And so when you consider overall architecture, gene content, and size differences, the human Y chromosome is only about 35% the same to chimpanzee. And so rather than going back and forth on all these technical details surrounding the methodology Dr. Tompkins is using to get the 84% similarity, you go right to the Y chromosome where there is no controversy. This is just a fact currently that the Y chromosomes are incredibly different. And so how do the evolutionists account for that? Notice this from the more recent paper, 2020, Dynamic Evolution of Great Ape Y Chromosomes. Despite a recent divergence of these species, their Y chromosomes differ enormously in size and gene. Notice the word enormously. They just put that in there, right? These aren't young Earth creationists. So a lot of times in debates, I would say to the evolutionists, it's not just 70% similar. It's actually 35% because the Y chromosome is only half the size between humans and chimpanzees. And I've actually had evolutionists say, oh, where do you get that? That's not true. Notice right here in their own literature, Matt, the chimpanzee Y is only half the size of the human Y. The percentage of gene families shared by these two chromosomes, 68% that split, is about 6 million years ago, is similar to that shared, notice this, shared by human and chicken autosomes that split 310 million years ago. Now, mind you, autosomal nuclear DNA is different than uniparentally inherited nuclear DNA like Y chromosome. But nonetheless, the illustration fits. They're basically saying this is how shocked they were. They were as shocked to see this as they would be, humans and chicken autosomes basically having that much dissimilarity. And so notice what they say here. Puzzlingly, so this is another example of a reality, some empirical evidence that was not predicted. And so they say in terms of shared genes and overall architecture, the human Y is more similar to the gorilla Y than to the chimpanzee Y. It's right there. You can't get away from it, even though human and chimpanzee have a more recent common ancestor. This is from the famous paper in 2010. The chimpanzee MSY, male-specific Y, basically, harbors twice as many massive palindromes as the human MSY. All you need is the Y chromosome. That is precluding data, preclusionary evidence, as some people like to say, in terms of the ancestry debate. Their arguments have not been tested. Oh, sperm competition, faster rates of gene conversion, faster mutation rates. There's problems with all of those explanations, okay? Yet it has lost large fractions of the MSY protein coding genes. Now, notice how they assume it's all been lost since the split, right, Matt? It's all been lost. And gene families present in the last common ancestor. Again, right from their own literature, surprisingly, more than 30% of chimpanzee MSY sequence has no homologous alignable counterpart in the human MSY. There's some people, even in some of the creationist camps, that say, there's no such thing as just a silver bullet or, just a fatal blow to evolution. I disagree. Mutation rate data, DNA function data, this Y chromosome data, they have not provided a sufficient evidence for why the Y chromosomes between humans and chimpanzees are so different. It wasn't predicted, but guess what? From the creation model, right, Matt? It makes sense. Humans were created separate and independent from chimpanzees. And so we'd expect there to be this kind of difference. But at the same time, even if it was more similar, like the 96% overall similarity, common design, but that's why this is differentiating, because the evolutionists were expecting a higher similarity. The fact that it's so different gives them a challenge, and it's a challenge that hasn't been met yet. What are the differences? You can go through this all yourself for sake of time. I won't. Again, you can go, here was a shock, 30%. Remember, 30% difference, but also half the size. So you're looking at basically a 35% similarity, which is very different. So this is their challenge. We've been asking this challenge for years now. They're now going to have to apply mathematical models to try to demonstrate how a sequence can change extremely rapidly, including wholesale rearrangement of significant parts and the evolution of entire gene families in a relatively short amount of time. You know, they always say, Matt, show us the math, show us the math, show us the math on the heat problem. Yeah, show us the math. Show us a sophisticated model on how these changes occurred, when they occurred. I want to see the details on this. This is going to be very difficult for them. I agree. Here's a 2020 article, commentary article on the 2020 paper I was talking about earlier. Notice what they say here, Matt. Our hypothesis is that the obviously they're not going to admit in these papers, well, you know what, there is some extreme discrepancies and differences here. So there's a good chance humans and chimpanzees aren't related. They're not going to get published. They're not going to get academic that'll be detrimental to their careers. And so they got to come up with some explanations, right? And so they say our hypothesis is that the accelerated change that we see in chimpanzees and bonobos could be related to their mating habits. In chimpanzees and bonobos, one female mates with multiple males during a single cycle. This leads to what we call, and here's the famous term, sperm competition. That's what we hear all the time from these guys, right? That's what Erica said as well. Notice how we think that this situation could provide, you can read it yourself, but this hypothesis, while consistent with their findings, they think they need some explanation, needs to be evaluated in subsequent studies. It hasn't been evaluated. It's not empirical. It's not scientific. It doesn't belong in a science textbook, because this is a pretty big discrepancy in the whole origins debate. And this discrepancy is on the side of the evolution side. And so, sure, you can have your story, you can have your untested hypothesis, but don't call it science. Matt, what do you think of their hypothesis anyways? This story that humans and chimpanzees are only 35% the same in the Y chromosome because of sperm competition. Mind you, Matt, as you think of what you would think of that rescue device, every single human male Y chromosome is what? Nearly identical, incredibly low genetic diversity. Every human male Y chromosome is uniform. And you can actually take every male Y chromosome and trace it back to a single male Y chromosomal ancestor in the not so distant past, about 4,500 years ago. But get this, the Y chromosome mutates fast. It's a rapidly mutating chromosome. It's about 3 per generation. So why in the world, if humans go back 100 to 200,000 years, why is there incredibly low genetic variation in the Y chromosome if it's mutating fast? The only way to explain a rapidly changing Y chromosome that also has low genetic variation population-wide is through young Earth creation, that the Y chromosome is young, it's not old. That's why it can accumulate a lot of mutations, but also population-wide comprise low genetic variation. And so if the evolutionary model were true, we should find some divergent Y chromosomes geographically, but we don't find that. We find Y chromosomes are uniform.
Especially in isolated people groups like the Aboriginals in Australia, right? They were supposedly isolated for 50,000 years, yet we came into contact with them. Their Y chromosome no different, and there was no speciation, and they weren't a different species. Breeding was identical. Yet we know from looking at animals that a speciation rate can happen as fast as 2 generations, and they can't interbreed. So what does that tell you? 50,000 years was not true at all.
Right. Yeah.
And hey, look at that. Their hypothesis is based on the fact that, we found something that we didn't mean to find. So when all these people in the chat are like, I don't know why that's surprising. Well, it was sure surprising to everyone else that found it. So it might not be surprising to you as a retro addiction, and you can pretend that it's not. But guess what? Every other scientist that believes in evolution, it was surprising to them.
But do you see how this puts them in the corner? I mean, I'm looking at the chat, and we love our critics. We love our critics. We appreciate your input. But I'm not seeing anything good. You know, I'm not seeing anything empirical. It's like this. excerpt from this article. It's a guesswork. Maybe this, maybe that. Joel Duff says, have you looked at the Y chromosome? From my understanding, the Y chromosome of Neanderthals, when they actually got pretty good sequencing data on the Neanderthal Y chromosome, didn't they discover that it's pretty similar to modern humans?
They didn't sequence the entire thing, but yeah, they found that the mutations are in the same place. But obviously, the similarity would be there because they branched off really early and then they died, and it's been thousands of years. But everything else matched up that they fell in line with human beings. Yeah, a matter of fact, they were more similar to us today than brown bears are to polar bears.
And it's not going to be anywhere near 35%. I mean,
right.
If the evolutionists can't even admit that a 35% similarity, that's what, 65% difference?
Yeah, crazy.
And to that, because remember how we say up, down, red, blue, like everything to them is evidence.
Everything fits.
And so here it's like, okay, isn't it funny how they never predict this stuff? But then, like with the chromosome 2 fusion, where we did a 3-hour video, it's so funny because they'll say, "Oh no, the highly degenerated nature of the so-called fusion is like that's all expected."
Yeah,
we'll point you to a couple different papers and so on. But it's like, wait a minute, in one of your own main chromosome 2 fusion papers, they're asking the question, "Why is the area so degenerate?" Why didn't they ask? Or why didn't they say the area is so degenerate, but it's what we expected?
Yeah.
No surprise here. Why are they always shocked? Why are they always surprised? And then typically your evolutionist apologists, especially on YouTube, they want to downplay the data, right? So in the papers, they'll oftentimes point out that this is shocking. This was a surprise. In the 2020 paper here, having to do with primate Y chromosomes, they said it's enormously different. But then you have your evolutionary apologists that just downplay it and say, wow, sperm competition. It's like, guys, you're going to have to do a little bit better than that.
That's pretty bad. When their arguments are always based on retrodictions and surprises, and then they have to come back and retrofit the data and do a U-turn and flip over and say, it doesn't matter. It just shows you that they don't care how many times it's been falsified, and they don't care that it's actually a failed prediction and that they had to retrofit the data yet again. They wait till the evidence comes in and then they backpedal. We don't have to do that. We've nailed it every single time. The fact that the critic doesn't know this or that they do and they don't care, it just goes to show you who we're dealing with, really.
Well, so think about this. You have this hypothetical split, right, Matt? Chimpanzees are our closest evolutionary relative or cousin. We go back, you see here, 6 million years, if you can see my cursor. So since then, you have your chimpanzee line going one way, and we would agree chimpanzees and bonobos, same kind. No surprise, bonobo and chimpanzee, Y chromosomes, even in light of this sperm competition and difference in mating habits, basically, they have pretty similar Y chromosomes, especially the gene content, right? And so they still stayed pretty uniform. And yet the human, I mean, these regions, like the heterochromatic regions here, is basically like almost non-existent in your chimpanzee regions. Your gorilla and human. So this makes sense. Notice how this fits perfectly with the separate ancestry model. Humans, their own created kind. Chimpanzees and bonobos, their own created kind. Even with the chimpanzee unusual mating habits, which probably is the same with bonobos, they still stay relatively the same. And they have a lot of diversity within their own groups. And yet still they're pretty close. And then gorillas, very dissimilar to both chimpanzees and bonobos. We'd say gorillas are their own kind. And then your orangutan as well, where you have a couple variations of orangutan, go back to an archetype. And so you got 4 created kinds here: humans, chimpanzee, bonobo created kind, gorilla, and orangutan. And the Y chromosome data fits perfectly. So anyways, think about this. Here's a brain exercise, I guess. So 6 million years ago, you have a split, right, Matt? Humans and chimpanzees. Human line goes one way, chimpanzee line goes the other. Well, within that 6 million years in the human line, what do you have? You got the Australopithecines, right? Australopithecus afarensis, Australopithecus anamensis, africanus. You've also got Ardipithecus, ramidus. Eventually, you'll have, according to their model, You'll have Sediba, Homo habilis, right? Their favorite transitional form, Erectus. Like, so if chimpanzees have these unusual mating habits basically today, well, what about all of the lineages leading up to modern humans? These ape-like Australopithecines, were they not having similar mating habits? Or, you know, how did they exist? I'm sure they existed in small groups and tribes, probably inbred. It doesn't make any sense. Why didn't the human Y also change a lot?
Right.
I guess is my it seems like they're cherry picking just the chimpanzee lineage and trying to say that all your major changes happened there, and your gorilla and human Y basically stayed mostly the same. But why? Just think about the evolutionary story and all of your different types that led up to humans today, including your Australopithecines, which probably had similar styles to some of your extant great apes. You know, speak to that a little bit, Matt. What are your thoughts?
Well, if they're going to invoke that a lot of these primitive ancestors that led up to human beings lived in these populations for millions of years, then we would expect to find that as fossil evidence as well. You can't say that humanity existed in this one region and they lived there for millions of years, diversifying and speciating, creating all these different things, and then not find any evidence for it, right? One of their first chains of the link is Sahelanthropus sagensis, and they found one skull. And so that was it. And I'm not saying that there weren't more of them. Obviously there were, but clearly, if they were doing anything along the lines of humans, where they would have been populating and growing in number and allowing the Y chromosome to stay and flourish and not diminish, then clearly we would be able to find more of that population. And we just don't. We don't find those millions of years of change, right? I think you got a super chat. I'm not really small here. I'll let you get it when you get back. So, matter of fact, that's one of the things that I think we're even going to get on, because I was shared a video last night that we're going to play in a little bit, and it looks kind of fun. It's just a YouTube short, and somebody mocking the idea of Adam and Eve, and that's going to be along the lines of what we're already talking about here, but nonetheless, I just don't see the story panning out with what they've said. I mean, where are the evidence of these millions of other species that have been living? They're not in the ground, and that's where I mean, think about all the money that they spent in time in Africa looking for these missing links, and how much time and they've investigated, and they don't find these things that they're supposed to. And then when they finally do, there's a division between scientists. There's lumpers and splitters, right? Some of them are like, they're not even in the human family tree at all. They're a completely different offset group. And then there's others who are like, no, they they want the money. They're like, they're basically in the human line. That's what they usually do when they first find it anyway. Especially people like Lee Berger. They're quick to jump on it and say, I found the next missing link, human ancestor. He makes the cover of all the magazines and he gets really popular. Then they find out later, oh, it was just a miniature human hobbit. Whoops. But nonetheless, it's already made its rounds. It's already made them the money. It's already made them more popular. So
okay,
politics,
Good stuff, Matt. So let's address some of these comments, and then we'll phase into your response to this YouTube short. So Zach B. Hancock says, mating systems are very different between chimps and humans. The latter are more like gorillas in their mating. Okay, we understand the argument. We've seen it, and I appreciate the comments. We've seen it in the responses from the evolutionists, the secular researchers on this, and they're saying, sure, it's untested. It's a hypothesis. We think this, but it needs to be evaluated further. Well, until it gets evaluated further, until you have something more definitive, more empirical, then at this point, it's just a story. Because you know what? 35% the same. There's going to have to be something more sophisticated than just mating systems. So let's move on to this super chat. Thank you so much. Appreciate the support. He says, about 65% of the human and chimp Ys are shared. That leaves about 10 million megabases of Y sequence that is unique to each species, 0.33%. So he's saying about 65% of the human and chimp Ys are shared. Well, again, when you consider overall gene content, architecture, size differences, you're only 35% the same. So that's about 65% difference. So I'm not too sure where you're, unless you're just focused, like Erica, I know at times she'll just try and focus on only the gene content, not care about the side. No, let's consider it all. And this is the difference that we find. Power Word says, have you studied the FOXP2 language gene? Yes, we have. Isn't that right, Matt? Yeah. Where are all the missing talking apes? It's funny how and doesn't Neanderthal have the same FOXP2 gene as us too?
Yeah, 2 of them. Yeah.
Yeah.
Yeah, we have 2. Chimpanzee, all primates really just have 1.
Redefined Living says, do we see genes being created as evolution would require, or are they breaking yeah, it's much easier to break something. And that's why where we see a lot of these orphan genes, these taxonomically restricted genes, we'll oftentimes find them. Let's take the felid kind, where we would say lions, tigers, house cats, cheetahs go back to a common ancestor. Well, since orphan genes are oftentimes species specific, you might find certain sets in, let's say, the lion genome, but not in the tiger. The evolutionist wants to say, well, because this is an example of one lineage gaining the orphan genes, right? But we would say no. It's a much better explanation that they're losing orphan genes over time. We've seen that in the fruit fly experiments, Matt. And so it's easier to break things down than to build something up, especially from scratch. And so, yeah, you have archetypes that come off the ark 4,500 years ago. They all have their respective sets of these taxonomically restricted and essential genes. And over time, through shifts in heterozygosity to homozygosity, you have breakaway populations. These populations become isolated. They become inbred, and you get new variations and you get speciation events. It's all a downhill process, though. It is all a loss of allelic variability and therefore a loss of what, Matt? Some orphan genes where one species in the same kind may have lost a set that another species has. And so that's a great question, Redefined Living. And I would say no, we don't see the uphill evolution of these genes or gene families or DNA elements. And so, okay, I feel like we had another Thank you for the super chat. He says, I admit evolution is true. Bubblegum Gun finally admitting that banana plants, tomatoes, humans, and chimpanzees are related through common ancestry. That's science. That's good. Finally, Bubblegum Gun is pro-science. For anybody unfamiliar with Bubblegum Gun, he's obviously being sarcastic. Return to fish, hashtag. I know Easy Myers would be all about that.
Yeah, he would.
Oh, okay. Apologetics 101, my brother. I highly recommend Brian's channel. Puts out some great interviews. He did an interview with Nathaniel Jensen recently, and he's also got one coming up with Dr. Tompkins. And so I am actually going to send a couple of these questions to Dr. Tompkins and get his response. And so, okay. So with that, I think we're caught up on questions. Let me see. Did you want me to play the short and then we'll get your response now? I just noticed the time. We've been going for a long time. So sure. Let's play the short and then we're going to get Matt's response. Okay, so here we go. Just let me know if the audio is working and we'll start now.
I am currently deep in the heart of Texas, but I want to take a second to answer this question. It's a really good question. The term mitochondrial Eve refers to the fact that we all get our mitochondria from our mothers. Sperm cells do have mitochondria, but they're only in the tails. And when the sperm and the egg fuse, the tail falls off. And so at the end of the day, all the mitochondria in your body came from your mother's egg cell. Now, similarly, there is a Y chromosomal atom because Y chromosomes can only exist in sperm cells because why would an egg cell ever have one? So this kind of perpetuates this idea that we came from two people, a Y chromosomal atom and a mitochondrial Eve. But no, that's not accurate at all. In fact, genetic analysis shows that those two people would have lived about 100,000 years apart. So no, it just doesn't make any sense. And this is why scientists should really stop using things like poetry and mythology to try to make things make sense, because while it is very pretty, it just ends up causing more confusion. So
lots of assertions in a 30-second video. Isn't that right, brother?
Yes, it is. That is a very simplistic way of looking at something. You can already tell he didn't dig deep, like, at all. Which is sad because it's an instructional video, right?
Right.
That's a science video, is what he was giving, a science lesson.
Many people are going to blindly believe that what he's saying is true.
Yeah.
And it's just assertions, claims. He's out of date, even just on his whole argument that mitochondrial Eve and Y chromosome Adam were separated by 100,000 years or so. And so why don't we, I want to say, remember a couple of years back we did like a 3-hour response to that PBS video?
Oh yeah.
That basically was a detailed attack on a literal Adam and Eve.
Right.
That was comprehensive. And so, to the chat, we've done full 3-hour shows refuting the critics on these points that Forrest brought up. And so we're going to respond to it now, but you can also get hours and hours worth of responses too. So Matt, did you want me to share your screen, brother?
Yeah, for sure. And while you're doing that, oh, it's already on there. Yeah, as you can see, I typed in, show me a picture of the evolutionary believer named Forrest, and this is what came up. I'm just messing around.
That's basically the kind of that's kind of the kind of research he's doing to come up with these arguments.
Unfortunately, if you actually Google mitochondrial Eve or Y chromosome Adam, that's what'll come up though. It'll only show you the evolutionary viewpoint from phylogenetic trees. It doesn't show you the actual empirical data that comes from something else, which we're going to get into in just a minute. But I did say 2 questions, and I'll answer them really quickly. One was, how many different kinds did you think were on the ark? I'll say a round number of easy, about 2,000. And another one is, how come an onion has a bigger genome? It's because they can't get up and walk around. They're stuck in the elements, and they need a larger genome to be able to survive the storms wherever they're planted in the ground. So they need that much bigger, though. So this guy that we're going to bash on, he's I checked out his channel. He is actually educated, and biology and genetics are his thing. So we are going to pick that apart real quick. Okay. So I just hope everyone can see. I might adjust a little bit to make it easier. Oh, that's helping a little bit. Okay. Here we go. So when he got those numbers, he was looking at this from the evolutionary standpoint only of a molecular clock that's based on the fossil record, meaning he assumes that there was a split between a human and primate about 7 to 7.5 million years ago. And you can see that they calibrate the molecular clocks to fit this. Okay. So we know that this is true because when we look at this, we can find here that the studies that measured mutation rates in human populations using pedigree studies have reported that the values differ significantly from rates that are phylogenetically compared to human and chimpanzees, meaning what we observe is not what we see based on evolutionary standards. Why? Here's a Wikipedia answer to that. Very, very simple. They look at the phylogenetic base, which is the assumed rate of a split, and they ignore the above version of the pedigree-based one, which is the empirical rate that we actually see when we look inside of humans. So therefore, they like going off of the assumed ancestry, and they look at and they make up their own rate based off of that. When we look at just some of the earlier studies, we can go back to Parsons, which was probably the 2nd or 3rd mutation rate in 1998 or 1997. 1997. He came out with a mutation rate and a substitution rate, looking at all of the different blood samples he had while working at the FBI. While doing this, he found that mitochondrial E lived about 6,500 years ago. And this became known as the empirical rate, and the FBI uses it today. So it has to be accurate. And what I mean by this is, if you decided to go out and kill somebody, and they came and they got the DNA, they said, okay, they come over to the house and they arrest you and your family, how do they know between whether or not you or your dad did it? Well, the fact is, your dad has a very similar to you, but he might not have as many mutations. You'll have more. So they'll look in the blood, they'll find that extra mutation that you have, and they'll guarantee that you are the murderer and not your dad. That's what gets usually fixed up right here. So what happens then is when they looked at O.J. Simpson's case, they found blood, but they found an extra mutation. And they said, well, it couldn't have been him. What they didn't realize is it was actually his son that probably did it. So off on a tangent on that. So what that means is Parsons found that there was one substitution every 300 to 600 generations, but it's not that's way too fast for evolution to be true. But there's also multiple other stories as well, and multiple other studies, I mean, and all of the studies get a most recent common ancestor just thousands of years ago. This is much faster than expected. 20% faster than expected. And when people challenge Parsons to this, when we specifically asked him, like, what's going on here, he said that his rate will be no problem. Okay. What he obtained, he would obtain no matter what, again and again. How do we know that? Because he actually did run the test again the next year, 1998, with a scientist named Holland. And Holland and him ran the numbers again, and they were drastically off the phylogenetic assumption rate based on evolution. They were way off, and again confirmed the earlier results, finding a slightly faster mutation rate, but it doesn't matter because, again, we're comparing the two and we're finding out which one is true. Is evolution true? Or when we look in human beings, are we finding young Earth creation is true? Again, we find this in multiple peer-reviewed studies that have nothing to do with one another, and they're always looking at the population of different groups of people, whether or not they're related, unrelated, it doesn't matter. We'll get into the specific studies. And as you can see, it's pretty much down the line. We're all getting our most recent common ancestor just a few thousand years ago, no matter where we look. Here was a lichen cave where they found a tooth about 4,000 years ago, the most recent common ancestor. We'll look in this one, which was a study that was done on a mathematical simulation. And they got, again, the most recent common ancestor just a few thousand years ago. So they wanted to say, well, maybe it was there's something they weren't accounting for, like heteroplasmy and stuff like that. That actually made matters worse for them. And then a lot of people say, well, Dr. Jensen, when he ran his numbers, he didn't take things into consideration like coalescence. Well, actually, he did take that into consideration. So that's out of the window as well. Then we'll hop over here and we'll look at this one, which is a hunter and gatherer southern one. It was inferred about this when we were looking about the association being limited between genetic samples, where they were looking at archaeological remains and trying to figure out, like, well, what's going on here? This is because what happens is what we talked about earlier, is that population sizes determine on when mutations reach fixation. And when they reach fixation is based on how many people there are. The smaller the amount, the easier it is to get the top of one, and therefore that mutation becomes fixed. So again, when we have a small population, bam, we get a mutation, and then pretty soon everybody has it. That's what haplogroups come from. We can also see here in this study that fixation is actually reached really quick, and so it was from genetic drift. And then when we look back at all the different studies, I listed them all here. We can go back to 1992 and then 1996, which is from Ludstorm, and then in Howell. He only ran females, but he got 200 to 300 generations ago. And then in 97, we get about 350 to 400 generations ago. You'll notice the common theme here, by the way. All of these are very recent outcomes when we look at the observed mutation rate versus the phylogenetic one. Dr. Nathaniel Jensen was at the bottom there. He ran his on a pedigree study that was done on 666 individuals, and then he made a prediction based on how many we would see based on that time frame and how many evolution would expect based on the same thing. Theirs was off by an astronomical amount. So we can see when we line these up, chimp versus human on the left, we've got everything from as low as 72,000 years ago to an upward amount of 300,000 years ago. That's a difference of 228,000 years, right? And yet every single pedigree study that's ever been done doesn't even come remotely close to their lowest estimate at 72,000 years. So then we can get into the Y chromosome. This one is even more amazing because the mitochondrial DNA is about 1,600 or 16,500 base pairs, right? So it's very small because the Y chromosome is almost 60 million base pairs. So it tells you a whole lot more of the story than just the mitochondria does. So knowing that, which one is going to be true? Are we going to look over and see evolution? Are we going to find this massive amount? Or are we going to find the young Earth creation example? Well, Dr. Nathaniel Jensen wanted to do the hard work, so he started reading pedigree studies that were done on the Y chromosome, and there were 4 of them. The first 2 that you see over here in 2009 and 2015 had a very, very weak look at the Y chromosome. They hadn't even actually counted all the differences. It wasn't until 2015 that Carmen et al. And his group decided we're going to sequence the entire Y chromosome using a more advanced technique. We're going to scan it multiple times and we're going to actually find out what's going on. And they did, and they found a high rate of mutations inside the Y chromosome, higher than ever before. And by doing so, they said, this isn't good. Basically, in a nutshell, they said this isn't good. These findings prompted us to explore additional filters, which means they don't like the outcome. They didn't like the results. They were way too fast. They were probably going to lose their grant money. So they published the answer on another section while ignoring the sections that had all the mutations in it. So one thing that was really good about that, though, is that while we looked at the Y chromosome, sorry, clicked on something wrong. We found out that this study right here gave the very most information of all, because they did what's known as high sequencing data, which means look at on the bar on the left showed all the mutations that were missed from the other studies, because what they did is they scanned the Y chromosome multiple times. They scanned it, and then they scanned it, and then they scanned it. And the more you scan it, the more mutations you find. So this is how many mutations were actually missed in the original studies. That's a lot of mutations. So Nathaniel Jensen said, we're looking at about 4,500 years worth of mutations. And that puts the evolutionary model of predicted timescale off by a dramatic amount. And so when we look inside there, what did we find? Well, we found about 3 mutations per person on average. Well, the average person has 440 mutations. That's just 146 generations ago. So that would place a person with a 30-year generation time about 4,380 years ago, or with a 35-year-old generation time 5,100 years ago. So Nathaniel Jensen placed the most recent common ancestor, Noah, being about 4,500, give or take, right in there, around the mid-range. This was actually more confirmation when we started looking at haplogroups, and he published this in his book called Traced, where he actually found that around the 6th generation, this person had 16 mutations, and then there was an explosion in all of the different haplogroups. And that aligns perfectly with scripture, showing that this is the generation of Peleg, when the languages were distributed. So that's amazing. So it's more confirmation that matches the biblical mutation, or the mutation rate that we would expect to find, and we did. And now for the curiosity example of all of these things. Human history only goes back about 5,000 years. And also, they publish these. These are in magazines, right? And then we can go around and we can see it. Geographical regions in Africa, about 5,000 years. We go back to the earliest pyramids, about 5,000 or 4,500 years ago. We found the Tower of Babel inscription. So, fossil paleontological evidence that existed as at least as a story. And then what do we find? We find about 5,000 years ago, civilizations everywhere started arising. They started picking up. And then here's another inscription of that. But yet when we look in Africa, what do we see? We only see a couple haplogroups there. But what would we expect to find if the biblical narrative was true and people were in Babel and they dispersed from there? We would think that that's where most of the mutations would arise. Look at that. That's exactly what we do see. This is the center of diversity for the expansion and explosion for all of the haplogroups that come out of that region of the world. Exactly what we should see. What about right here? Yeah, writing systems, same time. We go back about 5,000 years. Look at that. All of history right in that little window. But yet they want us to believe that 195,000 years of human history passed without anybody ever doing any of these things. And then bam, it's just an explosion right here. Look at this. Recent shows Indian migration from Australia. So much for the Aboriginals living there for 50 million years. Here we go. They're only 4,000 years from the new genetic data. How come they haven't speciated yet, right? Genetic drift would allow them living on that island, clearly diversifying and speciating. Without that amount of time, we would never be able to breed with them. And as I mentioned earlier, phenotypic diversity is the exact same between every race and group of people in the world. There's no hierarchical pattern there. That's exactly what we should see if all people are related right off the ark. And there's 3 groups of people coming from Ham, Shem, and Japheth's line. I put this in here, but that's too much to go over right now. Instead, we would see an African line being much more divergent because they had lived for much longer before we actually got out of Africa. Or if we diverged from Neanderthal hundreds of thousands of years earlier, then Caucasians would be much more different phenotypically. But that's not what we're seeing. So, oldest trees in the world only go back to, again, right around 5,000 years. We get the math goes back 5,000 years. What a shocker. I know, right? We get fossil record evidence of, what is this? Sorry. Historical records. We've got, how old's the oldest desert in the world, right? We're going back again. We got about 4,000 years ago. Sahara Desert starts popping up. Bam. How about genealogies? People have unbroken chains of genealogies going back in history, especially royalty, because it all mattered from blood, and their blood genealogies trace right back. This is the British King's List in the museum in Britain that shows a genealogy going right back to Noah himself, and those exist all over the place. This isn't random. And then we have flood stories and traditions that go back, and they all have a very consistent theme, and most all of them have 8 people that survive. And no matter where in the world you go, and you go to Cambodia, Guatemala, you go to Australia, you go to Africa, you go to Egypt, they have one thing in common. They have pyramids. And these pyramids are very unique because they all contain a very consistent theme. They're all actually built off using the same mathematical concepts of using the meter. Now, how would that be true unless we all came from a group of people that were using the same thing at Babel? You know, we look at ancient writing systems. They can be traced back again to the same story of biblical creation in writing systems. So even though the writings and languages were broken up, they diverged and they took these stories with them. How about human population growths? Human population growth fits right in with biblical creation. It doesn't match the evolutionary model yet again. These are all short things. Here's one. Mutation rates just randomly pop up 5,000 years ago. What a shocker. We have Mendel's accountant, right? What building is that in the background? Do you recognize that? That's right there. That's the NIH and the National Health Institute. Guess what they're using? They're using John Sanford's Mendel's accountant for biblical creation. Why don't they use some evolutionary model that talks about mutation spreading? It's because they're not true. They only care about what works. They don't care about creation versus evolution. Yet there they go. They use the young Earth creation model to do it. That tells you right there everything you need to know. And Mendel's accountant, if you want to see a little bit more on it, here's a study that you can find probably on Answers in Genesis. I highlighted it. You can also run the program yourself. You can implement your own starting situation. You can your base foundations of people. And here's an evolutionist that said it pretty well. I don't know if it's not going to play. I shouldn't have tried. We could just end off with this one then. We don't need evidence. We know it's true. That's what they believe about evolution. It doesn't matter how much I can point to things that don't match and how much inconsistency there is with their model. They've already made up their mind, okay? So I'm just saying, take another step back. Use statistical probability. What are the statistical probability that humans everywhere from every different group that there could have possibly been in the world, all waited 195,000 years just to come up with the exact same concepts, timing, math, writing systems, language, farming, agriculture, all decided to just build pyramids exactly the same around the world. It's hard work. Let's just do the same thing anyway. Let's use megalithic stones, because why not let's make it the hardest possible thing we can ever do. That totally makes sense, right? So I'm just saying that it's very peculiar that if young Earth creation is wrong, that everything seems to point to it. That's all I gotta say, man. I'm out.
That's all I gotta say. I'm out. That's the perfect way to wrap it up. And so, Forrest, if you feel like you can debunk that, we've had an outstanding evolution debate challenge for the last year and a half. So bring it on, especially the topic of ancestry, Matt, our main focus. Since we are over the 2-hour mark and we said we're going to do about 2 hours, and I'm kind of losing my voice and was out in the sun basically all day. So I'm going to wrap it up with a couple supporting points. Is my audio coming in okay, brother? Okay.
Yeah.
Yeah. Okay. All right, let me go through a couple of supporting points, and then I'll give a summary of basically what we talked about. Okay. So I just find it funny, his short there. It was obviously you can't be all that detailed in a short, but nonetheless, what do we have evidence for? We have evidence for, as you've pointed out, one Y chromosomal ancestor. We understand that just by levels of genetic variation. For example, here, and I won't get into the whole unrooted versus rooted arguments and all that. Let's just look at the raw data. Okay. Low variation overall in the Y chromosome. There's not thousands and thousands of mutations here. We understand the mutation rate is fast. Okay. This is literally these branches on the tree. They've got mutations in them. They're DNA differences. You can count them up. We know which mutation led to each branch. And the length of the branches on both the Y chromosome tree and also the mitochondrial DNA tree here are easy to explain in biblical time. And with a fast mutation rate, low genetic variation, there's no way for the evolutionists to take this back hundreds of thousands of years, specifically 100 to 200,000 years. Not with just a few. I mean, on the mitochondrial DNA tree, for example, you have on average 20 to 25 DNA differences separating any two people. Your longest branches, you've got what, maybe 140, 150 mutations, not a whole lot. And so it's only a little bit of variation you have to explain in just thousands of years. But the evolutionists, they have to explain, they looked at bottlenecks, genetic drift, so on and so forth, selection. And so with a fast mutation rate and low variation, as creationists, we can actually account for purifying selection as well and still have more than enough time. And so another thing to consider as well is that if we actually shared a common ancestor with the great apes and we split from a common ancestor with the chimpanzee 6 to 10 million years ago, Matt, well, we would have essentially split from a common ancestral population, right? And that population would have had a lot of genes floating around. Now, this ancestral population that we would have shared with the chimpanzees before the split would have had, therefore, a diversity of Y chromosomes. And so as that ancestral population basically diverged into different groups, for example, the human group, the chimpanzee group, or lineage, one group, let's say humans, could have comprised individuals that are more different to each other than they are to a chimpanzee on the Y chromosome. And this isn't the case. But the important thing is that it could have been if humans and chimpanzees really did split from a common ancestral gene pool. And so the point is, we have evidence for one male Y chromosomal ancestor of all people on the planet, and we have evidence for one mitochondrial DNA ancestor from all people on the planet. Now, we've also discovered Eve, and Dr. Rob Carter has published in the secular literature on this. And this has to do with the Eve consensus sequence. So you can check out this paper here, mitochondrial diversity within modern human populations. The worldwide human mitochondrial DNA consensus is composed of 16,569 nucleotides. Based on the current data set, 84.1% of the mitochondrial genome is invariant. And so the mitochondrial genome is this small piece of DNA that's only inherited on the mother's side, right? It's uniparentally inherited. Every generation you get mistakes, and you can look at those mutations. You could build a phylogenetic tree. And again, those branches all go back to a single woman. But the raw data that we have is that there's low variation. And since it's only about, it's 16,569 nucleotides, we can actually simply take all of the mitochondrial DNA in the world and line them up. This is basically what they did in this paper. And what they noticed was about 84 to 85% of the letters out of the 16,500 letters have the exact same letter. And so that actually tells us that Eve, the consensus sequence, basically they'll say, the evolutionists will say this is a coalesce sequence, where we would say, no, this is the actual sequence of Eve, because they don't have evidence for, other women, or they don't have evidence for, this single woman, but then again, other women that lived at that time. What we have is the raw data that tells us we have a sequence here. And this is the sequence of basically a consensus sequence of a woman that we've all derived from. And so basically, notice here, the world consensus sequence is unambiguous except for only a few nucleotide positions. The amount of mitochondrial sequence divergence within humans since the most recent common ancestor is quite low. Low genetic variation. On average, any two people or any person would differ from the consensus, from the Eve consensus, by about 22 nucleotides. That's nothing. There should be way more differences if we actually go back to, an ancestor 100 to 200,000 years ago. And so essentially, if we were to consider worldwide variability, we can see where everybody in the world, and this is what they did here, has an A, okay, ATCG. So every person has an A, but one person in the world has a T. Well, which letter did Eve have? This is how you come up with a consensus sequence. Well, Eve obviously would have had an A. If everybody in the world except for one person has an A, the one person has a T, well, then A would have been the original letter. And so just from this logic, you can literally derive this Eve sequence. We can know the mitochondrial genome. And we understand here, as you can see in the paper, that 85% of the mitochondrial genome is invariant. And most of the variations actually rare, which means it's found only in one people group or even one person. So what do we have, Matt? Low variation, a mutation rate that is fast. Most people in the world are only a few differences removed from the Eve consensus sequence, with obviously the African lineages being the most divergent. And so we know Eve. We literally have her sequence. And an illustration would be if I were to hand out 100 books to 100 different people and told them to copy that book, or let's say one book to 100 different people and have them copy that one book, 10 to 20 times. And then I get all the books, analyze them. You can see where the discrepancies are, right? You can see where the deviants are. And once you remove the deviants, you can find out what the original book said. And that's exactly what was done in this paper. Is they figured out, Dr. Rob Carter and Dr. John Sanford specifically, they figured out where what the Eve sequence looked like. And so again, this Forrest doesn't know what he's talking about. Pedigree-based mutation rates, 10-fold higher than phylogenetically derived mutation rates. So anyways, I'll wrap it up there because we could talk about this all day. But let me summarize what we talked about here. Okay. Basically, some critics, or even so some critics have misrepresented some of Dr. Tompkins' work and methodology on this. But there's also been some valid questions asked. And all we're wondering, right, Matt, since it doesn't really matter, as we're going to really focus on the Y chromosome, we're also going to focus on, as you can see here, I didn't really have too much time to get into it, but notice differences in gene expression. We identified 169 genes that exhibited expression different. So even in the same genetic sequences, you have differences between humans and chimps in gene regulation, gene expression, the way the genes are expressed, the way they're turned on, the way they're turned off. Notice this, our results indicate that the human, so even if the chimpanzee is 96% similar to humans, or if the chimpanzee is 84% similar to humans, it doesn't matter. There's still going to be a hierarchy, but there's differentiating lines of evidence that are more important to focus on anyways, like the Y chromosome, gene expression here, gene regulation. Notice this, this means even in the similar sequences, I can't emphasize this enough. Even in the similar sequences, there's still differences in the way they're expressed, in the way they're regulated. There's epigenetic differences, there's architecture different, there's differences in the 3D structure of the genomes. And so creationists shouldn't fear a 96% similarity. But if the similarity is only 84%, I mean, obviously that creates a huge problem for the evolutionist model. on top of all the other differentiating lines of evidence that we point to. But there seems to be a couple questions to sort through, such as, what would we get? Matt, let me know if you agree with me. Is it not a curious question? What would we get when we incorporate more species and use the methodology that Dr. Tompkins used to compare humans and chimps, but let's say humans to humans or chimps to chimps?
It's just going to broaden the spectrum everywhere.
So basically, is it a safer bet to just stick with the 96% similarity and then focus on the differentiating lines of evidence? Like, what's your opinion on it?
No, because I think we're going to find out that we share a lot more. There are human-specific things that they've never really tried to count that humans only have. You know, the orphan genes, the endogenous retroviruses that are specifically doing functional things, especially little what they would consider trivial function, right? Like what ENCODE found, transcription. Once we find that those are actually in a place where they're supposed to be because they're doing transcription factor, it'll be more for us. It'll be a lot more similarities.
So would you go currently with the 84% rather than the 96%?
Yeah.
Because, so up here, a couple points to consider.
Okay.
Richard Boggs, an evolutionary biologist, he acquired nearly the identical results to Dr. Tompkins using because before the chimp genome, it was humanized, right? They kind of used the human genome as a scaffold. And so there's some issues there. But there's been more sophisticated technologies now and analyses. And so Dr. Richard Boggs came up with a similar number to Dr. Tompkins. And Dr. Tompkins points out that if he included the unalignable parts, such as duplications, the genomes would be even more different, 80%. Now, Gutta Gibbon, in her latest video, she's pointed out that if she uses the same methodology that Dr. Tompkins uses to get the 84% number, she gets humans within humans, just comparing human to human at like somewhere between 85 and 90%. What are your thoughts on that?
I think she's trying to put the focus on trying to say that if there's a bigger difference than humans can't be related, then it shuts down the argument for young Earth creation. But it's not going to work, because we still, like I said, I think that the emphasis isn't really on this much genetic similarity, because we're not considering the similarity of nuclear DNA. Those are already differences that we would expect based on functional aspects. It's the differences that we're looking at specifically when it comes to the mitochondria and the Y chromosome that I say proves ancestry, because those are the mutations we can trace back to a common ancestor. We just can't go past that, even if we wanted. Even if we wanted to prove a common ancestor past the last bottleneck, we can't do it.
So you want to focus. So you want to focus on the bigger picture, the differentiating lines of evidence.
Right, exactly. I don't look at one thing. You notice I just gave a list of things. It was about mutation rates, right? All that was, my rant was literally starting with pedigree mutation rates. But you notice I went on about 15 different things, or 20. The reason why is because they all point to the same thing. It's looking at the bigger picture. I don't just hyper-focus and say, look, mutation rates prove that humans came about 6,000 so years ago. And then I never talk about anything else, because nothing else can point to it. I want to show everything that goes to it. So we don't just say, look at how similar we are, and then move on. That's what Tompkins did. Tompkins was like, look how different chimps are and look how similar we are. Boom, case closed. No, that's what he does in a study, because studies are focused on one thing. He's not trying to look at everything. He's doing one study on one thing. That's it.
Right. And so when you incorporate everything, so for example, here's a paper analyzing alternative splicing differences between humans and chimpanzees. And so the evolutionists, and I've heard, a lot of the more informed evolutionist responses to these differences is they're just going to have to invoke these changes came about after the split. And it's a lot of storytelling, like we see with the Y chromosome differences. And so notice this, surprisingly, 6% to 8% of profiled orthologous exons displayed pronounced splicing differences, level differences in the corresponding tissues from the two species. Again, in total, 1,526 exons, right? The protein coding regions versus your introns, non-protein coding intervening regions, basically. And exon sets from over 1,200 genes showed significant splicing differences among primates, epigenetic differences as well. There's differences in the human and chimpanzee brain genes that are great. They're big differences. We also found extensive species level divergence in patterns of DNA methylation. Not to mention the chimp genome is 10 to 12% larger than the human genome. And it's not in a near finished state like the human genome. It's more so like a rough draft, Dr. Tompkins' point is basically when you consider all the regions, just like Dr. Nathaniel Jensen points out, you can see it right in their own literature, that when you consider all of these regions, the differences are a lot more than if you want to just consider cherry-picked regions, basically. Earlier in DNA sequencing, years and years ago, they would take basically what they knew were similar genetic sequences and then compared those, and they'd get like a 98% number. Well, yeah, of course. Or like we pointed out earlier, if you're just going to focus on your protein coding regions as compared to the non-protein coding regions, then sure, you're going to get more similarity, because your non-protein coding regions, they're providing the metadata for the protein coding regions. There's a lot going on behind the scenes. And so I think it's the most sophisticated analysis to consider all the genomic features. And so there's some lingering questions there, but there's some differentiating lines of evidence, which is important for the viewer to understand that the evolutionists really struggle with. And I think that's basically the last thing I wanted to point out, Matt. So I think we actually got through everything we wanted to discuss, brother. It's been fun, been an interesting one, been a technical one, been some back and forth bouncing ideas off of each other, but that's fun. That's how we, proceed in this debate and better understanding these topics. So
yeah,
what's going on, brother? Any final words, final thoughts, anything else you wanted to get in there?
Maybe we could go live sometime and do a comments on comments for fun, and then another time we can go live and just do Q&A for the audience. That would be fun. But we have so much of this type of content. There's always a video out there we can just play and go at. That's always fun. Obviously, we need to do one with Joel Duff. I'm thinking genetic entropy.
Yeah.
He comes in here with some heat. He wants to bring it. Okay, let's do it. We'll play your genetic entropy video sometime. And,
we'll do. You know what? We're gonna do a comment on comments with Joel Duff. There's two questions. So Centurion, my question. He has a question: the polyfunctionality and polyconstrained characteristics of the DNA. If you or Matt Man can address this, if it's relevant. Yeah, real quick, the genome is polyfunctional. It's also polyconstrained. Basically, what that means is, we write books that can be read forwards, but books can't be read both forwards and backwards, or take every other letter in a book and you get another message or a whole nother story. DNA can do this. The DNA language, it's nested, it's multi-dimensional, it's chock full of meta information. And one sequence, you can have one message going forwards, one message going backwards, and a third message reading every other DNA letter. And so what that means is, if these DNA sequences that are polyfunctional, if they're hit with deleterious mutations, damaging mutations, it's polyconstrained, because it's only going to make that sequence or the message built into that sequence and encoded into that sequence worse. It's not going to get it better. You're only going to get the breaking down of DNA systems. Essentially, you're not going to build them up through deleterious mutations, because they're constrained. They're functional in so many ways that mutations actually could disrupt one mutation could disrupt multiple messages basically in a sequence. So Matt, I don't know if you had anything to add to that, but those are my thoughts, brother.
No, it's okay. We'll move on. What else we got? Where are all the gorillas and chimps? Awesome.
Patty Smith, Thank you for the $10 super chat. Where are all the chimp and gorilla fossils? Yeah, this was a question I asked Guts Gibbon in a discussion we had recently on the Australopithecines and the hominin fossil record. And so I asked, where are all the chimpanzee fossils? Where are all the gorilla fossils? These paleoanthropologists, they're always finding the so-called next big transitional fossil. But where, in the hominin line, right? The big findings would be in the hominin line. They want to find the human ancestors. But then there's a significant lack of chimpanzee fossils. Where's all their ancestors? Where's their fossils? Where's the evidence for them, basically? And that's where I argue that a lot of your extant great apes, their ancestors, those lines since the Ark, a lot of their bones, their isolated bones, have actually been mixed in with, the hominin collections. And so that's where sometimes you'll get your ape bones mixed in with your human bones. Obviously, evolutionary taxonomists would say that humans are human great apes, and your non-human great apes like chimpanzees, bonobos, they basically classify humans as apes. But so yeah, I think there's a lot of, and I have a book from Bernard Wood, and he points that out. There's not as much there's not enough reason or benefit to finding your chimp ancestors or your gorilla ancestors. Everybody wants to find the human ancestors, the big transitional form. That's where the money is. That's where the fame is. That's where the fortune is. That's where the incentive is. And so even some in the paleo anthropological community would agree with that. Now, the evolutionists, they'll say, right, Matt, they'll say, well, chimpanzees and gorillas, they lived in different environments that were not suitable for basically finding their fossils, finding their bones. Anyways, what are your thoughts, brother?
Yeah, I mean, they found the first chimpanzee bone. It was a tooth in 2005, right? That was their first discovery. But if we're talking about like human beings, we bury the dead. We kind of have, as far back as we go, Neanderthal, Denisovan, right? They found like literal burial mounds and graves. So we should be much more preserved. There should be massive amounts of being able to find these things if we literally live for hundreds and hundreds of thousands of years. And we're not just talking in Africa either, right? I mean, we're talking all over the regions, all over the place. And Neanderthal wasn't supposedly isolated to Africa. That was that was only a specific modern branch of human beings that were specifically isolated for 150,000 years there. So they would have still been burying their dead though. And we're not finding those hundreds of millions of people that should have been living in Africa doing those very things. So very strange.
Well, think about Homo erectus supposedly lived on the globe for 1 million plus years, according to the evolutionary model. Even the evolutionists would admit that they were pretty sophisticated hominin type, right? They could sail, they could make fire, they had campsites, so on and so forth. We would say they're fully human. Evolutionists would argue they're pre-human. But anyways, where's the evidence? If you just think about their generation time was similar to us, 25 years, and then they existed on the globe for 1 million plus years, there should be billions of dead bodies. Where are they? The most you're going to find it can basically be fit into into the back of a trunk. And so, okay. You know what? Since Gutsy Gibbons here, appreciate you being here. We were asking Joel Duff and Zach Hancock earlier, because I've gotten through about an hour and a half of your video, made a lot of notes, but I didn't note specifically the percentage using Dr. Tompkins' methodology on human to human comparison. The number you got, correct me if I'm wrong, was between 84 and 88%, if I'm not mistaken. We were talking about that earlier. And so anyways, we'll see if she responds. Which again, if anybody goes back and watches our presentation earlier, we've been going for almost 3 hours. I had a slide with questions basically for Dr. Tompkins to hopefully address regarding his methodology that goes outside my initial rebuttals and counters and so on to some of the things being said. But anyways, to the fossils question. Yeah, we're just taking audience questions here. So yeah, so my question would be, where's all the evidence for erectus existing for that long? Where's their campsites? Where's, all the evidence for their bones if you've got billions and billions of dead bodies and so on and so forth? And so, I think that's a legit question. And it just doesn't seem like we have much evidence for them existing that long. Matt, go ahead. Any thoughts, brother?
Oh, she responded. She said it depends on which Tompkins method you're referring to, 11, 12, 13, 15, 16, 18. And she said that was the end of her video. I don't know if you got through that whole video, though. Did you?
No, well, I got about an hour and a half into it.
Yeah, but she
And
said
then-
at the end there was that's where she did something. She asked group.
2018, I think, would be the most up-to-date one, obviously. And so I
Got it.
think, yeah.
And redefine living going after base theory. Like nonstop in the comments.
Redefine living in base theory, best buds.
Every time I look over, boom.
Well, we've got a Matt Man and base theory debate coming up in in June. So again, Matt, you're going to be busy. This goes back basically 2.5 hours ago to our initial Gutsick says, I asked Tompkins questions at the end of my video and Roop's interview, and he did not answer them to answer your question. Okay. But I remember seeing in your video, you had numbers where you had said you had run the same DNA sequencing that Tompkins did using his methodology on humans to humans and basically got a low number, if I'm not mistaken. But anyways, Grayson's coming at you, Matt. So those are fighting words. It's gonna be a wild June in the debate dojo.
Nice one.
And so, you know what? I think we basically touched on everything that we wanted to in the last 3 hours. I was up early and then also out in the sun for a greater portion of the day. So I think I'm basically ready to wrap things up. We've answered all the audience questions and we've touched on the few different topics that we wanted to, brother. So any last words, thoughts, comments, arguments?
No. I think it was good. It was good. We went a long time. I mean, we always try to aim for an hour and a half or 2. We never make it.
Well, it's funny because remember we were going to go live. We were going to do it last night, but then we realized, you know what? We're going to try and fit all of this within an hour. And so it's probably best we save it for tomorrow night. And then guts it, Kevin. You should have came 2.5 hours ago. We were having fun with our favorite critics, Joel Duff. We were having fun with Dr. Zach Hancock. Erica wants to tell us or me about her human chromosome 2 work that's coming up. Oh,
good.
She's giving me nightmares before the big release. Well, I am looking forward to that one. I'd like to see your response for sure to the chromosome 9 data. So she says in the 2011 to 2010, the Han Japanese human was 79 to 87%. In the human 21, about 84. Okay. Okay, here's the one I was looking for. So in the 2016 to 2018 was 99 human to human, 98 human to chimp. And so I'm assuming she's saying using Dr. Tompkins' same methodology, his same approach to the sequencing. That is what she...
So looking only at human chromosome 21 gave more of a discrepancy than looking at the, I'm guessing the entire protein coding region to get the 99.
If I'm not mistaken, I was reading an article that talked about how they did do some sequencing years ago on a specific chromosome, 21 or 22, that they knew beforehand anyways had a lot of similarity. And so when they came back with about a 98% similarity, it wasn't really shocking because that was one of the most similar chromosomes anyways between humans and chimpanzees. And so basically, I'll go to my slides here again real quick. we started the video responding to Dr. Marcus Ross's criticisms and pointed out how Dr. Tompkins' analysis was basically well, let me see here. His estimate was based on alignable regions. All unaligned regions, including large indels, were excluded from his percentage estimate. And so basically our questions, my specific question was, if we're looking at quote unquote an insertion deletion, or we're looking at a duplication, you got the chimp genome, you got the human genome, and you've got this gap that is an insertion, or let's just say a deletion. Why are we assuming that it is a deletion? Or if it's a duplicated gene, why are we assuming it is a duplicated gene? It's the evolutionist model that would assume basically after the split, humans go one way, chimpanzees go one way. The chimpanzee lineage is going to have its own mutational events. The human lineage is going to have its own mutational events. And so I can see why the evolutionists would say, well, there's this gap here, right in between these two genomic regions. But there's something present in the human genome. So it must be a deletion. And if it's a deletion of, let's say, 1,000 base pairs, should we weight that, or should we count that as 1,000 differences, or just one event? Or if it's a duplicated gene comprising, again, 1,000 base pairs, do we count that as 1,000 base pairs, or do we count that as one event? But I think the underlying question is going to be, well, I think we got to determine firstly if this really is a duplication, or if this really is a deletion or an insertion. And that's going to be based on two different starting points. One who assumes humans and chimpanzees go back to a common ancestor, and another model that say humans and chimpanzees were created separately and basically don't go back to a common ancestor. I was shared screen, so I can't really see what was being said. Any thoughts, Matt, as I look through the comments here?
No, it's just like, I guess we could just go back to the fact that it would be nice if Tompkins would reply more, so we could get to the bottom of like what he's working on. He's very hard to contact. Remember, our first initial ones was through ResearchGate. 6 months later, I didn't even get a reply. So I don't know why this guy's so hard to get a hold of. A lot of people are easier, but even then, you might just get like a vague response.
Well, I think that's why it was so good to, it's been a blessing to do a lot of work with Chris Root behind the scenes. And so through him, we were able to look up the chromosome 9 data. We were able to look for other instances in the genome where these telomeric calling cards, TTA, GGG, CCC, TAA, are found in that head-to-head organization. And so we did that without Dr. Tompkins' help. And when he was shown those findings, he was pleasantly surprised. He was like, wow, this is pretty awesome. So Gutsa Gibbon says Tompkins ungapped parameters. Okay, yeah, that's what I gathered, is that is the major issue that the critics are having with Dr. Tompkins' work. This is different from how we treat indels, insertions, deletions. Those are separate problems. And so I think it was both for the first hour of the video, that was basically what we were responding to, was Dr. Ross's criticisms involving the indels, duplications, his illustration there, with one paper versus 4. What do we count that as, right? So my lingering questions are basically this. Let me share screen again. I am looking forward to that chromosome 2 fusion work, because that is one of my main focuses right now. Okay, so right here. Lingering questions. So we chatted about this for quite a while. What happens if we compare other organisms using the same methodology as Dr. Tompkins, chimps to chimps, rats to mice? Matt, would you say rats and mice go back to the same created kind, or separate kinds?
Ooh, I've never looked at those actually. maybe different because they kind of carry different pathological diseases. They also have different chromosome fusions that branched out. But that could happen from isolation, like the Mendelian mouse that has like almost no chromosomes left. But yeah, if they can break down that quick, I'd really have to look. That's a good one. Usually unclean animals were only brought on in pairs, so they wouldn't have as much diversity in them that we would see in the cleaner animals. I can look into that one though. That'd be good.
Yeah, no, that's so basically, if I'm representing the critics' argument accurately, if we were to go with the 84% number, right, Matt, between humans and chimpanzees,
yeah,
and say 84%, that's too different for evolution to be true. There's no way for the evolutionists to account for 300 to 400 million differences in 6 to 10 million years since the split. And so the critics will go to two different species that we would say go back to the same created kind, let's say rats and mice, or maybe lions and tigers, and say, but hey, rats and mice, they have about an 83% difference. But you say they go back to the same created kind. And so basically they're saying our argument is inconsistent.
I see. Based on differences that we can find within humans, if they're too different from one another. Well, again, that's really not how we would trace, because those differences, we would have to be able to gather how fast these differences are occurring. How often are deletions happening? How often are these gaps being created? And right now, our best way to trace lineages is through mutation, and finding them on the Y chromosome, going backwards in time. That's the best way for real ancestry to be tested back on a paternity test, right? So if we're just looking at overall gaps in differences, and then we look in human beings, it's kind of like, well, how did those gaps get there? Were they already there? I mean, what are these differences because of what reasons, right? If we're looking at an epigenetic thing and they're off, and it's not being seen because the sequence isn't even activated because it's an epigenetic regulation, it's going to look different. So we got to there's a lot of considerations. Does the person have less telomeres because they're 90 years old? And then when they're testing a 2-year-old, they have more telomeres? Well, think about that, right? You lose chromosomes each time. You lose telomeres at the end of your chromosomes every time there's a cycle. So if you test an old person to a young person, they're already genetically different. If you test your wife to yourself, she has no Y chromosome. They're 58 million base pairs difference. So there'll be more. The more you look at, the more differences there's going to be. It's just how quickly can those differences arise? We would have to do the math on
that. Yeah, good response. And so Gutsy Gibbons says, every question on that slide I answered in my video in gratuitous detail. Yes, I saw those portions. And so that's why I wanted clarification on the exact numbers. And therefore, those questions pertaining to Dr. Tompkins' methodology are questions I'd like to see Jeff respond to, because Matt, we spent a lot of time basically talking about what number do we go with, the 96% or the 84%? And we want to be as consistent as possible as we focus on what we believe are differentiating lines of evidence when it comes to DNA similarity. That's why I spent a lot of time on the Y chromosome. That's why I spent a lot of time on gene expression, gene regulation, epigenetics, so on and so forth. And so we're happy to go with a 96% number, as we pointed out for a while in the chat, if that's the more consistent number to go with. And I think the criticisms of Dr. Tompkins' methodology currently being advanced by the critics, I think it's, and tell me if you agree with me or not, Matt, I think it's Jeff's job to address those, since that's his work, that's his methodology.
Well, yeah, I mean, we can answer for him, but it might not be the answer that he wants, and he has to go
guessing,
back and just damage control for us, because we see that all the time. We see Christians jump up and give crazy answers. Remember when Erica debated G-Man? What in the world? So then we got to go back and say no. He didn't know what she's talking about. There's the actual answer. So it's the same thing. He doesn't want to watch our video and say like, no, these guys answer that completely how I didn't want to. So we just got to hear from them. That's all.
Right. And I feel at this point, with the way that I know the way I specifically argue, and the arguments I utilize, they can either have the 84% similarity argument as supporting evidence, basically, or not. Like either way, I feel like it's not going to hurt me. I'm okay, as you said, to go with the 96% number, because regardless, there's going to be a hierarchy. The reality of a hierarchy in the biological world exists. Whether it's an 84% number or a 96% number, we're still the most similar to chimpanzees. And so whether 84% or 96%, I still want to engage the critics on what I believe, and they'll obviously disagree, and that's fine. That's where we discuss these issues, where I believe the differentiating evidence lies. The Y chromosome dissimilarity, for example, which I spent a lot of time on. And so my point here with these questions is I'd like to see Dr. Tompkins address these in the same way Erica has. And people like you and I, Matt, we can kind of sit there and examine the way both sides, Erica and Jeff in this circumstance, answer this. And we could decide for ourselves as young Earth creationists, taking our own approaches to engaging the ancestry topic, and see if we want to go with the 84% number or the 96% number. Does that make sense? And is that kind of where you're at too, Matt?
Yeah, I just want more information. With Nathaniel Jensen, it was much easier because his work was everywhere. If you wanted to message him, you can go on Telegram or
he
Twitter.
Answers the next day, basically, on Facebook, right?
Yeah, he's right there. So it was really nice. And I'd be like, okay, well, let's take your mutation rate. It's have you considered this and consider that? He goes, yes, and here's why. But with Tompkins, man, he might as well be living in a cave somewhere. I don't know if he's just working all the time, or does he just hate socialization? We don't know. I just know that he doesn't respond. So it might take more time, unfortunately. But you've got your own work now on the chromosome too. So it's good.
Right.
You're doing your own thing. I'm doing mine. We're all each doing our thing.
And that's why, especially when it comes to endogenous retroviruses, which is basically my main focus these days, and currently chromosome 2 fusion with the work Chris Roop and I are doing, I get a ton of questions coming in through Facebook. I get a lot of questions coming in through email. I really like the Facebook questions because then I can just click the audio button and give the questioners, 5-6 minute audio response.
Wow.
Saves from the carpal tunnel, right, Matt? We're always talking over audio on WhatsApp all day. And so we do our best to defend the work we're personally doing. And if we have specific methodologies that we're employing in our studies, we would definitely want to defend those. So these were basically my responses to the indel arguments, or the arguments that appeared to be saying Dr. Tompkins was basically going with a maximum number, when in fact he was going with a minimum, because he did not include unalignable parts such as large duplications. And so Dr. Tompkins would argue that it would be even more different if he included those. But critics like Erica, it's the ungapped criticism, I think, that is the main focus, but also the consistency of the models. So I don't know. We'll see if we got any more questions. We're coming at 3 hours. We'll probably wrap it up here. Appreciate you joining in, Erica. $2 super sticker. Thank you very much. All right. That's good. This has been a fun one because, let's see, bubblegum. I can use the method that puts chimps at 84%, no problem. And Erica can't complain about it. That's true, bubblegum. He's a fixity of species kind of guy, right, Matt? Chihuahuas were created in their current form. So
dang.
We'll give you that, bubblegum gun. Your polytheistic model, I guess you can. Guts and Gibbon said it's actually not a maximum/minimum number. That's what I thought it would be, but it doesn't even scale. Like human/chimps are more similar than human/human. So the scaling is off. Apparently, if you're going to get human, if you're going to get instances where human and chimps are more similar than human, I mean, that's pretty bizarre, since we know humans are 99.99% the same. Low variation, that's a reality. And so to stick with, right, using Dr. Tompkins' ungapped methods, Erica's saying. So, these are criticisms that we would love for Dr. Tompkins to engage.
We'll invite him on. So we'll see if he comes on. How about that?
There we go. Let's
just invite him on the channel. He's taking interviews. He's on other channels. Why wouldn't he come here? I mean, send out the invite. He goes on channels where they get 100 views. Come on, come over here, get 1,000.
We'll have him on to address, or at least engage his thoughts with Erica. Now, the Logos RA presentation was a lot of fun. I was pointing out earlier that I was going to host that one. I'll be hosting either the next one or the one after that. But we had Chris Roop host it, which made sense. I had a show that same day and everything. And it was a good turnout. The critics were there, had some good questions. I had a question of my own. And so this is how we advance. And you and I, we just want to know, do we utilize the 84% number or the 96% number? Because either way works with the arguments we use anyways and the approach we take. And so that's basically been our point all along, as we believe there are differentiating lines of evidence that are more important to focus on. Gut6 says, you can tell Tompkins. I think it has something to do as well with his parameter. Max target sequences. Something is askew in his published parameters, even outside the ungapped thing. Okay, well, I'll be curious to see where this leads. I've had a lot of fun tonight, I should say. I've gone through everything I wanted to, gave my thoughts. We bounced ideas off each other. Matt had some good discussion and a lot of good questions that came in. So let me remind everybody, though, as we do move into next week, we've got another several debates. Tomorrow, for you guys in the States, it's a holiday. And so we have a Memorial Night debate on dispensationalism. So Pastor Matt first, Charles Jennings. That'll be tomorrow at 9. And Gutsy Gibbon, I look forward to your video on chromosome 2 fusion. I will definitely give you my thoughts and counters to that. And so, okay. Matt, any last words, final thoughts, brother?
No, man, we got 3 hours in. I'm good.
3 hours, we're ready for bed. So thanks everybody for tuning in. We'll see you tomorrow night, and it's been a good one. God bless all.